Oui, est-ce qu'on m'entend? Ok, ça marche. Est-ce qu'en régie, c'est bon au niveau des caméras? Sur YouTube, je vois uniquement une caméra de fond. Est-ce que c'est possible d'en mettre une?
Ça va venir.
D'accord.
Il y a un léger décalage.
D'accord. Oui. Et du coup, oui. Maintenant. Lorraine, donc tu restes là pour le temps d'introduction, pour la modération YouTube. Bon. Depuis ton ordi? D'accord. Ok. Ah. Bon, on va pouvoir démarrer. So we're gonna start. Just please have a seat. Make yourself comfortable. And I would just say hello to everyone in the room and online. We are connected with YouTube on streaming too. So we have another 100 of participants online. And just a few words of introduction to open these 2 days of Arcad Scientific meetings. So we are pleased to welcome you, so many of you, for the Arcad Scientific meetings under the title Crop Diversity at the Core of Transitions. So these 2 days are organized on the occasion of the inauguration of the new Arcad Biological Resource Center located on Agropolis campus in Montpellier, under the supervision of INRAE and CIRAD. Today will be entirely devoted to the scientific seminar, through which we wish to share with you the themes and challenges addressed by the Arcad scientific community. To do so, we've designed a program in 3 parts. Maybe some of you have the programs printed in hand. So first, from 9 to 9:30, we'll propose you to go back over the history and logic of the Arcad project. Then, from 9:30 to 10:20, we'll lay the information of the scientific, environmental, and social issues of agrobiodiversity with the help of 7 keynote speakers from diverse profiles, representative of international organizations, biological resource centers, research centers, and farmers' organizations. Finally, and above all, from 10:20 to midday, and throughout 3 sessions, the latest knowledge available on crop diversity, and highlight the role that crop diversity has played in past transitions, and the role it must play for today's and tomorrow's transitions. In addition, please note that an exhibition of posters presenting the work of the research teams involved in Arcad Center will be at your disposal in the main hall during the coffee and lunch breaks. For the 100 listeners following us online on the YouTube channel, we invite you not to hesitate to address your questions to the speakers through the chat, which will be moderated in live by our team. I now would like to give the floor to Claire Billot, head of the Arcad Institute, who will present you the logic of these meetings. Without further delay, and in the name of the organizing committee, I welcome you to these 2 days of Arcad scientific meetings on crop diversity. Thank you very much. Claire.
Thank you, Alexandre, for this introduction. So, we, as a research community involving mainly in the organization two research units, AGAP Institute, from which I'm a representative, and DIAD unit, from which you can get in touch with Yves Vigoureux in the room here. We are very pleased to invite you to participate to this scientific meeting of Arcad, which takes place in Montpellier during these 2 days, today and tomorrow. We are honored by your presence, whether at distance because of the particular sanitary conditions, or face-to-face here in Montpellier. This meeting is very special for us. This is the first one of what we hope will be a long series. It marks the inauguration of the Arcad building, whose official opening will take place next week on Thursday. And it is open to a wide community, widely than the usual scientific research community. Arcad stands for Agropolis Resource Center for Crop Conservation, Adaptation and Diversity, which has a strong focus on Mediterranean and tropical plants. It's an open platform for better knowledge and management of Mediterranean and tropical crops agrobiodiversity. It has been initiated via an Agropolis Foundation research project some 10 years ago, and it holds multiple and open scientific ambitions, of which one of the material manifestation is this building that you will visit tomorrow morning. It holds an state-of-art equipment largely dedicated to the conservation and characterization of cultivated plants. These days are meanwhile an opportunity to pay tribute to the few people who have largely supported this project. Some of them are in the room here, and some of them are also online. But Arcad is also a manifestation of shared time, for which these 2 days are a presentation. And in these 2 days, as a research community, we hope to share our orientation and our work, but also take the opportunity to revisit our objectives in regard with contribution of other actors. In these terms, Arcad goes far beyond ex situ conservation and encompasses more than just the researcher housed in the building. We hope that it will reflect the desire for diverse opening that we have and that we believe is necessary to answer the situations of evolution of agriculture, climate change, demographic pressure, global change as a whole. The diversity of cultivated plants is for us at the heart of a portfolio of solutions for these rapidly evolving agrosystems in relation with this global change and the accelerated transition we are experiencing. To ensure resilience, sustainability of our food system considered as a whole, nutrition quality, multiple uses of plants, diversity is a solution. Diversity of plants is constantly evolving, and we all actively participate in its renewal. It is just thus not only about the diversity of cultivated plants, but also the diversity of paths, the diversity of applications, the diversity of agriculture and societies, the diversity of practices, the diversity of point of view that we need to address. And these diversities have to be implemented. These 2 days will present our scientific ambition, which we want to be in close communication with the social evolutions. The days have just been designed as a way to draw up an interdisciplinary overview of the knowledge of crop diversity. We have structured them in several stages, adopting a chronological view, but also opening for more crop diversity and for transition. So these days will be introduced by repositioning Arcad in its function of diversity conservation in a related international initiative. This will be this morning. Then the scientific pathways, as have been described by Alexandre, will go on for the end of the morning and the afternoon. We will open the doors of the building for visits of the facilities tomorrow morning, and open the doors, but also the thought on various needs, wishes around plant agrobiodiversity by multi-actor workshops and proposals tomorrow afternoon. And finally, we'll end up with the points of view of the institutes supervising our units, AGAP Institute and DIAD. But before that, it seemed important to us to put the dynamic in context by giving the floor to the Agropolis Foundation, which has largely contributed to the first steps of this initiative and goes on with supporting us as a research community. And follow this presentation by one of the people who carried the project from a more historical point of view, Jean-Louis Pham. Let's have time for presentations, fruitful discussions, sharing and opening new pathways for a more inclusive research. Thank you very much.
Thank you.
We are trying to solve some technical problems. And Marie-Christine Cormier-Salem, the directrice of Agropolis Foundation, will then give her talk. Actually, we don't manage to share the presentation. Yes, it is. Okay, Marie-Christine, the floor.
Thank you. Dear colleagues, as director of Agropolis Foundation, it's an honor and pleasure to welcome you for these 2 scientific days. And following what said Claire Villot, I would like first to emphasize the originality of Arcad. Arcad program includes 3 components or pillars: a scientific program, infrastructure and equipment. I will let Jean-Louis Pham introduce the 2 last pillars, and we'll focus the attention on the research program supported by Agropolis Foundation from the beginning. First of all, I will remind you briefly the missions and actions of Agropolis Foundation, and then show in what extent and why Arcad is a flagship program for the foundation. Who we are. Agropolis Foundation is a facilitator and a funding instrument at the service of its charter members, 5 charter members, a public institution of research IRD, CIRAD, INRAE, and 2 public institutions of training, Institut Agro Montpellier Supagro and University of Montpellier, at the service of its scientific network and its partners. It is located in Montpellier, and it is a private scientific cooperation foundation. Here you have the introduction of the scientific networks supported by the French ANR, the LabEx Agro, with more than 4,000 agents, 160 scientists, 40 units. I have underlined the units involved in the Arcad program and distributed according to the main domains. But let's say that, for instance, DIAD, AGAP, GAFL, and PVBMT, for instance, there are also social scientists involved in these units, and it's very important, as underlined by Claire. The ambition of the program was to be really interdisciplinary, to focus on interdisciplinary and to foster the synergies between our communities. Our objectives are 4. First, to support the scientific dynamic, promoting interdisciplinarity and integration from field to fork and from local to global levels. Reinforce the reputation and attractiveness of the Foundation's network to contribute to international scientific cooperation program. 3, facilitate partnerships between the scientific network and non-academic stakeholders. And 4, mobilize resource and funding. With two-fold approach, we have elaborated a new scientific strategies focused on 3 axes: agriculture and climate change, agriculture and biodiversity, responsible production and consumption, and Arcad respond totally to the second axis, agro-biodiversity. And you are here the different domains who are encouraged to work together to address the nexus of these objectives. What we do? We try to mobilize excellent and relevant science. We launch calls for proposal and commission projects designed to enhance the creation, sharing, and use of knowledge, and to build synergies to greater impact. We continuously seek to establish partnerships with public and private organizations, and to recognize and reward talent. We organize international scientific prizes. What are our means? From the beginning, in 2007, we got an initial budget of 20 million euros. And then in 2011, we got a fund for LabEx Agro of 25 million euros, and LabEx Agro has been renewed, and we got a new fund of 12.5 million euros till 2024. A few data on Agropolis Foundation to better situate the importance of Arcad program in the frame of the foundation. We have funded more than 485 projects of research, training, higher education, conferences, and innovation. 955 fellows of 79 nationalities, 546 scientific publications, and we have more than 420 partners in 80 countries. But we do not do alone. We have also international partnerships, national institution, regional institution, for instance, in Africa, award, African Women on Agricultural Research and Development, and we are also a member of networks of foundation with whom we share the same values of resilience, inclusivity, equity. And here you have the distribution of co-funders of the foundation. Coming back to the Arcad program, I would like to show you in what way, yes, in what extent it is a flagship program and has had a leverage effect on the activities of our scientific community. I go very quickly because is not my field, and Claire already has reminded us that Arcad was focused on the notion of domestication, in fact, on the beginning, to study the history and patterns of crop domestication and adaptation at various scales, to studies of evolutionary genomics, population genetics, and social sciences. And there is a focus on Africa and the Mediterranean basin. Here you are some figures, the main data or key data on Arcad program, Scientifique Arcad. The amount is really significant for the foundation, 3 million euros between 2009 and 2015, 30 partners all around the world, and especially in Guinea, Kenya, and Morocco. 4 LabEx agro research units involved in the project, and a project lead by Agap and Diad, 41 staff recruited, especially 20 master students from France, Angola, Brazil, Benin, England, Germany, Mali, Senegal, and more than 58 scientific papers. And Arcad has really a leverage effect. And in terms of research, training, and innovation, you are here, the events supported by the foundation, Arcad Scientific Days. In terms of training, the training component was really crucial. And here you have an illustration with a second international course, Agrobiodiversity, People and Plants, organized with Moroccan partners and Biodiversity International. There are also, in the frame of the Open Science School 2016, 2 projects supported by the foundation. One on the diversity of wild banana in Southeast Asia, and Arbopolis on diversity of safu in Cameroon and a rural-urban gradient. But there also, we have supported also mobility. There are 2 also new projects issued from Arcad with an enlarger team and approach, Genome Harvest, which is the development of the bioinformatics components of Arcad, and COEX for the production of new knowledge on the diversity of seed and their uses by farmers. And more recently, the Seed Attach project, which is an ongoing program with really, as the title say, the willingness to focus on the attachment of all the actors to the seed in Africa. I am very proud to be here today because I think that this support, 3 million euros, is commensurate with the ambitions of this research program. On the other side, Arcad meets our strategic priorities, notably the axis agrobiodiversity, our preferred approaches, and our values, which I will summarize in 3 I: interdisciplinarity, inclusivity or intersectoriality between academic and non-academic actors, and international. During these 2 days, I hope for fruitful exchanges and for transforming perspectives in the face of current global and local challenges. Thank you.
Yeah, we need some help. Okay. Where are the files? Ça, après, je vous ai pas pour oui.
Okay, thank you very much. Good morning, everyone. So I'm very pleased to open these scientific meetings today and to tell you a little bit about the genesis and objectives of Arcad, which, with many others, I contributed to develop. So today, as mentioned by Alex and Claire, we're here in connection with the inauguration of with the official opening of the Arcad building. Actually, the Arcad building was delivered 2 years ago already. But the history of Arcad actually can be tracked to 2007, even for first ideas of developing an initiative on agrobiodiversity can be tracked long back away. So in 2007, there has been a note by the region Languedoc-Roussillon about the possible development of an initiative to create a new biological resource center devoted to genetic resources of tropical crops. Within these 14 years, there has been a number of events. We observed a number of logo changes, which is quite a good indicator of the passing of time. We started with the building that was supposed to be built here, and then we moved the place, and finally it's here. The first idea was to have a round building, but as you know, the building is more closer to a square shape. This is to say that a number of things happened over the last 14 years. There has been ups and downs. As Marie-Christine reminded us, the funding of the Arcad Scientific project in 2009 was really a key milestone to bring credibility to the whole idea. Then we got important support from the EU in 2013, and also from the state and the region in 2015. Overall, we got a lot of support, and I think from these many institutions, and I think today we have to thank these institutions for their support, and this makes us accountable to succeed in the mission we have been entrusted with. Also, I take this opportunity to thank the many people, and I cannot mention all of them, who contributed to the project. Just want to mention my colleague Jean-Pierre Labouisse, who is in the room, and who I thank him for his support during the scientific project. And also, of course, my colleague Jean-Marie Prosperi, who is online, I think. And Jean-Marie was really a key person in the development of the building program. So this initiative, Arcad, is a pluri-institutional one. The idea was to take advantage of the wealth of diversity of collections we had in our cold rooms in Montpellier, but also on the wealth of expertise of networks we had developed over the last decades. And the idea, as already reminded by Claire, was to develop, and this is really the starting point, and the mission we have was to develop an open platform for genetic and genomic resource conservation, analysis, research, and training. This is focused mainly on Mediterranean and tropical crops, including crop wild relatives. We say that Arcad is a resource center. It's not only a biological resource center, it's also a center for technological resources, methodological resources, knowledge resources, and training resources. Today, with the opening of the building, we are able to bring together many components. First, a biological resource center called GAMET, which brings together collections of rice, sorghum, fonio, maize, medicago, cotton. around this center for biological resources, there are facilities, automated storage, cryopreservation, seed phenotyping, like the DNA lab, also an information system, and they contribute to the good functioning of the biological resource center, but also they are very helpful and they are very important for all the research teams which are associated to Arcad and to the networks which are worked into the dynamics together with the research teams. And finally, as mentioned by Marie-Christine, Arcad has an ambition in terms of education and training. So to make the objectives very short, we could say that Arcad scientists have the objective to understand how crop diversity has been shaped by environmental and anthropogenic pressures. The goal of this research are to design and to nurture approaches to foster the use of biodiversity of Mediterranean crops and tropical crops. The very ambition of Arcad is not only to develop the scientific objectives, but also to boost synergies between disciplines, between teams, between projects, between stakeholders on crop biodiversity. A way to say that is, here is a picture of a wall painting in the Arcad building. And on the left-hand side, you can see something that was designed by the artist as being a representation of molecules. But we could also say with a different view that this represents seed networks. And this is the way we have to work in Arcad. We have to cross the way to see things, and we have to cross approaches. One of the missions, of course, is to make the platform as open as possible and to connect with the regional, national, and international actors. That will be the role of my colleagues in the next session to develop these aspects and to see how Arcad can be integrated into the various dynamics. But let me say that, as Marie-Christine said already, that we have to involve both academic and non-academic communities. I guess you know that IPBES designed this figure, showing that to have an effect about the drivers that affect biodiversity on Earth, we have to mobilize a collective energy and to mobilize multi-actor interventions in order to have levers on the way we can change the things, and in particular in our field, to change the way agriculture affect and manage biodiversity. This is something that we started already a little bit, and that will be developed during a workshop tomorrow afternoon. Thank you very much, and good luck, Arcad.
So thank you very much. So following this presentation of Arcad and this welcome speeches, we wanted to have also a series of speeches of selected distinguished people representing a selection of a diversity of organization involved in crop diversity study, management, and regulation at the international, at the national, and local levels. And we've asked each of them to give us a very short speech to present themselves, of course, and their organization, but also to tell us how they see Arcad within this existing institutional landscape, and if relevant, their expectation vis-à-vis such a multifunctional and open platform that combines in one single place, as Jean-Louis has presented, a different function as like conservation, research, and training. So this breadth of perspective will give us a good overview of the diversity of type of organizations and perspective with whom Arcad is already interacting or will have to interact in the future. So we'll have first Mr. Kent Nadozi, the Secretary the Secretary of the International Treaty on Plant Genetic Resources for Food and Agriculture, based in Rome, who will give us a global policy perspective on plant genetic diversity, followed by Dr. Stefan Schmitz, the Executive Director of the Global Crop Diversity Trust, an organization that is dedicated to support conservation efforts of crop diversity at the global level. And then we will follow with 3 representatives of organizations intervening at the national level, 2 representatives from the French national organization. The first, Audrey Didier, in charge of the coordination of conservation efforts at the institutional level in France, and the other one, Michel Tixier-Boichard, representing a dedicated infrastructure of biological resources for research and innovation in agriculture. The third one, Njido Khan, represents a national agronomic research center in one regional excellence center on dryland cereal, Le Serras, located in Thiès, Senegal. And finally, we will conclude with 2 local organizations, one in France called the Conservatoire Botanique Méditerranéen, involved in the in-situ conservation of cultivated and wild Mediterranean plants and trees, and represented by Mr. James Molina. And finally, Mr. Mahamadou Hassan Sanoussi, who represents a federation of farmers organization from Niger, involved in several research projects on crop diversity with partners worldwide, including Arcad researchers. So I'll give the floor to Mr. Kent Nadozi, who should be online with us. Kent, the floor is yours.
Yeah, thank you so much, Salim, for that introduction, and thank you to the organizers. I think the list you read out now, and given the wide scope and spectrum of participants to this event, I think it's indicative of the role that Arcad is going to play in the coming years, and the broad range of stakeholders that will be involved in its activities. So dear colleagues at Arcad, ladies and gentlemen, I'm in fact very grateful for the invitation to this scientific meeting that is preceding the inauguration of the Arcad Center. Unfortunately, due to the current restrictions on travel, I cannot be with you in person, but I'm pleased to interact with you online. The fact that crop diversity has historically been a fundamental asset for the development of agriculture is well known and proven. So is the fact that today we feed the world with plants that are the result of plant genetic diversity conservation and utilization. That is why we have an international agreement for states to recognize their interdependence and cooperation on these matters. And that is the International Treaty on Plant Genetic Resources for Food and Agriculture. And as Salim said, my name is Kent Nadozie. I am the secretary of this international treaty. Equally well known is the role of human beings, that is farmers and breeders, in creating, maintaining, and improving crops that feed us all. Plant genetic resources are utilized as the building block for crop varieties that are adapted to different needs, which includes higher yields, as well as adapting to new and emerging challenges, both biotic and abiotic stresses. However, with the divergent visions of food production and resource management, with technology allowing for the design of genetic improvements through sequencing, some people wonder whether the actual preservation of crop utilization and utilization of crop diversity is changing its ontology and needs profound realignment with the technological realities of today. Therefore, I welcome Arcad as a valuable partner to reinvigorate the projection of crop diversity into the future of sustainable production of food in the context of ecological transition. Multilateral institutions for valuing the traditional resources, for instance, those in gene banks and farmers' fields, will remain essential to position crop diversity in the future of diversified food systems. As recent scientific evidence produced within the international treaty demonstrates, Countries remain highly interconnected in regard to the primary regions of crop diversity important in their national food supplies and production systems. Countries highly depend on crops whose genetic diversity largely originates from outside their borders. The dependence of countries on crops from other countries and regions has increased over time, and this is associated with diverse food supplies and production systems, as well as GDP. So the rationale of the international treaty and its system of global exchange of crop germplasm is still very relevant. In this context, recognizing global interdependency needs to be matched with the capacity at all levels, global, national, and local, to use the resources. To achieve this goal, the international treaty needs research and implementation partnerships. As the COVID crisis has shown us and has highlighted that resilience is fundamental and nested in systems that enable the deployment of crop diversity. As noted in the introduction to this science meeting, as much as diversity is constantly shaped by human societies, the interactions within those societies are also very fundamental. At present, such interactions in the realm of global crop diversity management are not exactly frictionless. By way of example, science and policy seem to move away from each other, or science moving much faster than policy. And diverse actors are often seen as expressing polarized positions. Sometimes science is also viewed only as plant science, and diverse scientific perspectives, for example, those in social sciences, are considered peripheral. In this respect, thanks to the supporting network of institutions such as CIRAD and its AGAP research units, I am convinced that Arcad is uniquely positioned to bring the very much needed inclusiveness of stakeholders in crop diversity management. Concretely, Arcad will not only support technological platforms and conservation facilities, it will also launch multidisciplinary research activities on crop diversity and involve in these activities all sectors concerned, including civil society, developing country research institutions, and farmers' representatives. This inclusive approach will certainly be beneficial to advance our common value propositions centered around sustainability and equity. As Arcad reconnects conservation with research and training in collaboration with a diversity of stakeholders, it will strengthen the discourse around crop diversity and help align it with current challenges. Finally, French institutions have been animating processes and actions under the international treaty since its adoption. I am certain that this collaboration will continue to be mutually beneficial under the framework of Arcad. Thank you so much for your attention.
productivity of millions. Hello, I'm Stefan Schmitz. I'm the Executive Director of the Crop Trust. Unfortunately, I cannot be with you today in person, but I'm so thankful to Arcad for giving me the opportunity to address you virtually. First of all, let me congratulate you to this new resource center for agrobiodiversity. This is a real achievement. This is real innovation for food security and for agrobiodiversity. Food systems are vulnerable and fragile. They are so interdependent. We need to find solutions together. We are facing two main challenges. First, we need to feed more with less. More people with better nutritious food by using less land and with less environmental impact. We need to practice agriculture within planetary boundaries. At the same time, we need to increase productivity of millions and millions of smallholder agriculture, improve their nutrition, their income, their livelihood. That would be a real driver for development of the global south. A lot needs to be done to address this issue. But I can say one thing for sure, we need better seeds. Against this, I'm delighted to see initiatives like this you are presenting here and celebrating here today. This new resource center for agrobiodiversity. It brings together the brightest of minds to find solutions, to develop creative solutions to secure our children's future sustainably. Crop diversity is a key foundation for food supply. It is a global public good, a global asset we need to invest in, and we need to invest more. We need better seeds to be able to deal with climate change in an appropriate way. We need more resistant crops. We need new varieties that are more resistant to higher temperatures, to flooding, new pests and diseases. But plant breeding is not simple. It is, for example, research intensive. So we need to put our heads together, investment, smart brains and everything to create an environment for making all that happen. Crop diversity plays a key role, and the Crop Trust plays a key role in this. We support the global system of ex situ conservation. Today, there are 7.4 million accessions in gene banks worldwide. And this is 1.4 million more than 10 years ago. We do not have more accessions secured in gene banks today. They are safer secured. They are duplicated. Most of them have duplicates now in Svalbard, the global seed vault up there in the Arctic. Important collections are held by the CGIAR, the Global Agricultural Research Network. CGIAR is our most important partner. A good start has been made, but it's not enough. Our North Arc of the 21st century is not yet full. More need to embark. The Crop Trust helps to raise funds, bringing data together, identify gaps, duplicate for safety. We support emergency operations, and we work together with key partners in this global system of ex situ conservation. International cooperation is key here. International cooperation at many different levels. We need to work together to raise funds. We need to work together, enter into a global dialogue to find the right policy solutions. We need to work together in the implementation and in research. Strong partners are needed. We need a strong voice for the conservation and use of plant genetic resources. We need to create excellent knowledge. And Arcad here is an excellent example of that. Again, let me congratulate you on the opening of this new center. Enjoy the conference. Be well. Thank you very much.
Good morning. My name is Audrey Didier. I am the National Coordinator for the Conservation of Plant Genetic Resource. And I will first of all thank all the organizers to give me the opportunity to present the National Collection, which was set up in 2016 by the Ministry of Agriculture and has two complementary elements. The first one is an coordination structure within GEVES. And the other one is the Okay, as I said, the national coordination, so we have a coordination structure within GEVES. And the other point is the consultative commission, which brings together representatives of 5 ministries. For example, Ministry of Culture as PGR, our heritage resource. But it also brings some research, public research institutes. Some of people are in the room today. Some breeders, associations, collection managers, and people involved in the use and promotion of PGR. The national coordination has several objectives to identify and support the stakeholder involved in the conservation and characterization of plant genetic resource. A specific procedure was set up in 2019 of official recognition of collection manager by the French government. And since 2017, a financial support from the Ministry of Agriculture helped to embody 37 projects aiming to ensure or improve the conservation of PGR collection for over 500,000 euros. Then is also to identify the pool of resources which have an importance to France, the so-called national collection. And this collection currently gathers 5,062 resources of 9 groups of species, including crop right relatives. The objective is to improve the long-term conservation and sustainable use of this resource, and this concerns different kind of aspects, as of course quality of conservation, but also knowledge about the maintained PGR, characterization and know-how, the knowledge about national and international regulation, and of course the access to the resource and to the associated data. On this point, Arcad is already involved in the development of the portal, as Jean-Louis Pham said, Florilège, which gives access to the PGR collection maintained by the institutes, the public institutes. Technical infrastructure developed into Arcad can also help in different ways to improve the conservation of PGR, as for example by offering safe duplicate rooms to other collections, and or through studies on the impact of conservation on genetic diversity by helping to question the current conservation practice and genetic diversity to preserve it better. Since the 90s, France has chosen to rely on network for PGR management, And if this organization remains current, it is necessary to enhance it with the diversity of stakeholders involved in the management and sustainable use of PGR. This also includes the development of interaction between the different kind of PGR conservation and management to create a continuum between the management, the in-situ management and ex-situ conservation. In this respect, through the convergence of expertise and the approach developed for networking academic and non-academic stakeholders, the Arcad initiative can help to better understand the social, environmental, and genetic aspects to be integrated to implement for the implementation of a continuum, which that is beneficial to all, and that allows better preservation and sustainable use of genetic diversity. And I thank you for your attention.
Good morning. I would like to thank the organizers of this scientific event for giving me the opportunity to present to you the French infrastructure of biological resources for research and innovation in agriculture. Genetic resources are at the crossroads of major challenges for science and society. Erosion of biodiversity, as reported by FAO and IBIS, as well as the renewal of agriculture systems with a push for agroecology and a new role for genetic diversity. In addition, the Nagoya Protocol is now regulating the access to genetic resources and the sharing of benefits from their uses. Besides, genomics is at the origin of a strong technology push for genetic resources and their characterization until their modification with genome editing. And these challenges are relevant for a large range of diversity, particularly in agriculture, with plants, forestry, animals, and microorganisms. So we need to secure, document, and provide access to genetic resources. And this is the mission of the RAR research infrastructure registered on the French roadmap. This infrastructure gathers genetic, genomic, and biological resources collected or produced by research in agriculture, food, and environment for temperate or tropical conditions. It is organized in 5 pillars with a common steering committee and a scientific advisory board. You will see here the 5 pillars according to the biological domain or the type of researches, microorganism here, crop of major interest for this seminar, forest, domestic animals, environment. And you can see here that various entities, scientific institutions are supporting these pillars. Altogether, the infrastructure gathers 32 biological resource centers with more than 800 users each year. And I would like to remind that by biological resource, we mean a biological material and its associated data. If we take here a quick look at the collections, depending on the pillar, you will see that for microorganism, the infrastructure hosts more than 20,000 strains. For crop diversity, there are more than 200,000 varieties. Forest, about 10,000 varieties. Domestic animals, 70,000 animals. And for environmental research, 15,000 soil samples, 1 million insects, as well as fish scale collections, and samples relevant to characterize the environment. So in types of samples, we have germplasm with reproductive potential, tissues, whole organisms, DNA bank, and back libraries. You will find more information on our website with a general portal, which makes possible the discovery of collections. And this portal also provides links to each pillar for placing requests for resources. There are more than 100 scientific papers published each year associating the different BRCs, relating to genetic diversity, population dynamics, gene mapping. The infrastructure brings support to BRC for data management, quality management, ABS implementation, and business model in order to optimize their operations and increase their visibility and recognition. Arcad is a very important component of RAR. It will secure long-term conservation for plant species of strategic importance, tropical species, maize, medicago as a model plant. It is also interested in supporting advanced characterization of genetic diversity thanks to a well-developed research partnership. So you will see in this seminar the importance of crop genetic resources for research and development in agriculture, and the infrastructure RAR is highly supportive of the science using crop genetic diversity. And I'm very happy now to hear a scientific discussion about crop genetic resources. Thank you for your attention.
So thank you, Michel. As you can see, we are juggling with pre-recorded video, with the Zoom live presentation, and with in-person presentation. So that's, I guess, the new normal in our new environment. And we'll have now Njido Khan, who very nicely, while traveling today, has recorded this morning his presentation and just sent it to us. So thank you very much, Njido. He's in Burkina Faso and traveled from Senegal to Burkina Faso today. Thank you, Njido.
Merci. Good morning, everybody, and thank you to the organizer for the given opportunity to share some views. Je m'excuse de ne pas être. We see Arcad as a collaborative instrument, an international device of infrastructure and scientific resources, generating knowledge that could and should fit public and private decision makers to halt the loss of diversity. We also see Arcad in an interdisciplinary perspective, including key actors and civil society that have complementary knowledge to engage in transformative approaches. In Senegal, this view of community engagement is translated in a clear commitment of the government to the conservation of biodiversity through national, African, and international treaty and conventions. Some limits was made. That's why the Senegalese governments have launched in 2015 a new strategy and action plan for the conservation of biodiversity. And the vision by 2030 is to restore, conserve, and harness biodiversity in order to provide goods and services in a sustainable manner with an equitable sharing and benefit and advantage in order to contribute to the economical and social development. In his operationalization in this strategy, the Senegalese Institute of Agricultural Research, ISRA, is a major player in the action plan through the management of seeds and the associated knowledge international gene banks. Like a biological resources center, ISRA has set up a system made of satellites for the conservation of plants, forests, microbial, animal, and fishery resources. So his regional center of crop improvement, CERAS, recognized as Center of Excellence by ECOWAS, ISRA extends his mandate to the sub-regional level as a leader in the management strategy of genetic resources, research, training, and dissemination of agricultural innovation on dryland crops. Some efforts to slow the loss of biodiversity, however, require a little more commitment and rigor in the application of law and governance in favor to biodiversity conservation, supported by behavior change and national coordination, bilateral and regional. Initiatives for sustainable solutions for the preservation of biodiversity have been taken, such as the ECOWAS Forest Convention Plan, the Tree Convention of Party on Climate Change, Deforestation and Biodiversity, the International Tropical Timber Agreement, and others. Some are being implemented also, such as the Green World, the Great Green World, from the Sahel to the Sahara, which aim to restore 100 million acres of degraded land by 2030. Also, BioDev 2030 project, which is an experimental approach that integrates biodiversity into the developmental of 13 countries in Africa with a diverse socio-economics and environmental context. More recently, some studies here conducted by the DIAD team in collaboration with us also shows that if in the case of PubMed, for example, there is a need for regional cross-border and multi-stakeholders collaboration to reduce the vulnerability of the biodiversity of the species to the climate change expected by 2050. The challenge of biodiversity must be taken into account in developmental policies, both public and local. The loss of biodiversity is above all a sum of local events. Local community must indeed become more involved by integrating biodiversity issue into their developmental action plan, but also encourage local population to apply measures for the preservation and rational use of their ecosystems. How to recognize respect of ecosystem and beneficial but sustainable use of resources. So thank you very much. This is some views I would like to share with you, and thank you very much.
Thank you very much, Njido, and so we'll finish with two presentations. I think one is in person here, Ms. Jem Molina from the Conservatoire Botanique, and then with Mohamed Sanoussi from Niger, a pre-recorded video.
So thank you for the organizer to invite me to this colloquium. I'm very honored. My name is James Molina. We used to say Jam in French. I work in a National Botanical Mediterranean Conservatory, which is, I'm sorry, I'm going to read my paper, which is involved in conservation of wild plants. This institution is held in Porquerolles. It's a little island in the south of France near Toulon. We are more or less 12 conservatories in France and forming a network, including all French territories, Corsica, and the ultramarine departments. To understand our mission and the reason why we're interested in a collaboration with Arcad, I will briefly decline them. We have three main topics. First one is knowledge and survey of wild plants, including their geolocalization, to be shared with public institutions, managers of natural spaces, searchers, and so on. We can share something than 5 million data of plants only in the French continental Mediterranean region. We also work in situ and ex situ conservation and expertise for administration collectivities about protected plants, endemics, and also plant of interest in a conservation way. Initially, we were only involved in wild and rare and threatened plants, and step by step, we extended our activities to common plants, to invasive plants, and more recently to the wild crops relatives, whose conservation, as you know, is threatened by the global changes, particularly in the Mediterranean region, which is, as you know, a hotspot of biodiversity. For us, that is quite a revolution to come from endemics in the highest mountain and typical natural habitats to the commonest human-dependent plants. We also work since a few time in genetics, making phylogeny and phylogeographic studies, and we need support of searchers to help us to do those studies. I have to add just a precision. We yet participate to Arcad with Flow Research that is mentioned, with our collection of olive trees, national collection of olive trees, which are in Porquerolles. We do possess seed banks. They are very dispersed all over the French territory. They are of different size, different technology, and subject to different hazards, as power outage or equipment failure, and not entirely secure. The partnership with Arcad will permit three main progress for us by sharing those technologies. The first is to secure for long-term conservation a double of our seed banks of the rarest plants of France. And the second one is to be known at national or international level and can be able to make seed exchanges in a facilitated way. By now, where it was only manual, it can be more automatically with Arcad. The third is to develop common research on topics of conservation dealing with wild and cultivated plants. Particularly the wild crops relatives were ignored by us up to now. But recently, a decree published in the official journal of the French Republic defined the missions of the botanical conservatories, and the genetic results are expressly notified. So this opened a new way, I should say a new opportunity for us to explore this main domain in conservation and research. And just for conclude, we have been contributors at a little scale for the Arcad projects, which grew up to be now a reality, and we hope to be collaborators in a shared partnership for a long time, I'm sure, and I hope. I thank you for your attention.
Many thanks, Mr. Molina, and we'll finish with a presentation by Mohamed Sanoussi, which will be in French. Sorry for the English speakers.
Le groupement paysan du Niger, FGK Morgan, est une organisation paysanne créée en 1993 avec 5 unions. Le groupement paysan du Niger, FGK Morgan, est une organisation paysanne créée en 1993 avec 5 unions. La date d'aujourd'hui, elle compte 29 unions membres avec 1524 groupements à la base, totalisant plus de 57 000 producteurs et paysans du Niger. FGK Morgan est une organisation paysanne créée en 1993 avec 5 unions. La date d'aujourd'hui, elle compte 29 unions membres avec 1524 groupements à la base, totalisant plus de 57 000 producteurs et productrices, membres physiques dont 63% des femmes. Elle intervient dans 3 régions du Niger, la région de Dosso, la région de Telabéri et la région de Niamey dans sa partie périurbaine. Morgan a développé une stratégie basée sur l'agroécologie. Elle en promeut les principes et la mise en œuvre auprès des unions membres dans toutes ces activités, qu'il s'agisse de l'appui à la production, de la transformation ou de la commercialisation. Morgan œuvre aussi dans le cadre de la gestion et de l'utilisation de la diversité cultivée, en promouvant l'usage des diversités de variétés adaptées à la diversité des solutions de ses membres. À ce titre, la Fédération a activement participé à la mise en œuvre du projet COEX et collabore avec plusieurs institutions de recherche au Niger, l'INRA, l'ICRISAT, l'Université de Niamey, l'Université de Telabéri, l'Université de Maradi et le CIRAD. Les activités de recherche-action menées depuis une quinzaine d'années sur cette question visent d'une part à conserver la diversité cultivée et d'autre part à co-construire avec la recherche les outils permettant aux paysans de choisir les variétés qu'ils vont cultiver en toute connaissance de cause par rapport à leurs besoins et aux problèmes qu'ils rencontrent dans leur vie de producteurs. À Morgan, nous sommes convaincus que la présence d'espèces collectées en Afrique de l'Ouest est conservée au sein d'Arcad, plus la présence des chercheurs de différentes disciplines comme évoqué plus haut, travaillant déjà avec Morgan, et la possibilité de formation offre des perspectives de poursuite et permettent un approfondissement de la collaboration déjà entamée. C'est pourquoi nos besoins et nos attentes vis-à-vis d'Arcad sont les suivantes. Dans le domaine de la conservation, l'admission de nouvelles variétés cultivées en sus de celles qui y sont déjà et un appui permettant à la Fédération maurienne de disposer de son propre système de conservation. Dans le domaine de la recherche, la poursuite de la recherche-action dans le domaine de la diversité cultivée, et dans le domaine de la formation, le renforcement des capacités des producteurs chargés de la gestion de diversité cultivée au niveau des unions. De plus, il nous semble légitime que des fédérations telles que la nôtre puissent aussi participer de manière un peu formelle aux réflexions sur les orientations des activités d'Arcad.
So thank you to all the speakers of this opening speech, and some of them in a very difficult condition. So a very warm thank you to all of you. And I think we will now make a short break before moving to the core scientific session with a series of different presentation breakout in 3 different sessions. So thank you very much. I think we will resume at what time? 10:40. So we'll have a short break outside there. There, yeah.
And we can see, no, it doesn't work. Yeah, just please enjoy the poster exhibition. Thank you for all the 7 keynote speakers. They presented the wide spectrum of academic and non-academic partners of Arcad, or potential partners of Arcad, and we just take the 15-minute breaks. You can enjoy the poster exhibition in the main hall, and the coffee served for all of you. So thank you, and see you in 15 minutes.
Okay, so welcome everyone. It's a pleasure to welcome you all in the room and online for this scientific seminar of the Arcad meetings. As you will see, today we decided not to present the research that we are doing here, with one exception. And to get an insight on this, you can have a look at the posters that are displayed in the hall downstairs. Instead, we took the opportunity of this seminar to invite researchers from outside Arcad to feed our curiosity and also maybe to plant seeds for future collaborations. So the seminar, the aim of the seminar today is to draw a panorama of crop diversity in many of its dimensions, genetic, phenotypic, but also sociological, ethnological, and ecological. The seminar will be organized in 3 sessions, 1 this morning and 2 this afternoon, and it will be a sort of chronological order. So we will start with a first session on past transitions, where we will look at domestication as an example of transition. Then we will examine how diversity can be mobilized to respond to current transitions, mostly with the climate change. And finally, this afternoon, we will discuss some leads for the future. So without taking too much time from the speakers, I will introduce the first session. So I'm happy to welcome our first speaker, Matthias Jakobsen from Uppsala University in Sweden. And Matthias will take us through the early developments of agriculture and the spread of agriculture in human populations. Switch to the Zoom. So Matthias is going to be talking from Uppsala. Matthias, you can share your screen. We can't hear you. Okay, we can see your screen. Can you speak? I forgot to mention that we'll have a few minutes for the questions at the end, around 10 minutes of questions if we're not running too late. And we will try to combine questions from the room as well as questions online. And together with Helen, we will moderate the questions at the end.
Sorry, we cannot hear you.
Yes.
Matthias, it should work now. No, we can't hear you. I think it worked.
Let's see if there's a difference now. Do you hear me now?
We can hear you very loud, but we can hear you.
Okay, thank you.
Okay, thank you.
Okay, good. I'm glad that you hear me, and thank you for the introduction and thank you for the invitation. I hope you also can see my slides and that you can adjust the volume if it's too loud there. So what I will do is to walk you through some of the results that have come out in maybe the last 10 years of digging into humans and digging into the human genome, and in particular past human genomes, in trying to understand what happened during the Neolithic transition. So what we're really going to focus on is humans, but I have a small catch at the end to bring back the importance of plants into this. So let's start with the Neolithic transition. See. Get the next slide. Okay. So my plan here is to talk a little bit about the Neolithic, the new way of life. Then I'll take a detour into Scandinavia, partly because that's where most of my research has been done for this time period, but also because that's where these patterns actually became apparent first of what I'll be talking about, that there are two distinct groups meeting and mixing in Europe. Then we'll zoom back out to the European arena, and then we'll see a little bit about where does these farmers come from and who were the people who were migrating. So let's start with some well-known facts from archaeology. So it's quite clear from the archaeological record that something happens across the European continent, maybe starting about 10,000 years ago and finishing perhaps 5,000 years ago. So if you look at the archaeological record, there's new artifacts showing up, including pottery, and maybe pottery is the most well-described artifact that suddenly starts appearing in the southeast of Europe, or already in the Middle East and in Anatolia, and then it spreads across the European continent towards the north and towards the west. These artifacts have been associated with a lot of other things, and they are often known as going under the name of the Neolithic package. There's other things than just pottery. So, for instance, there are new ways of using the resources of nature. So there's domesticated animal remains associated with these archaeological sites, domestic animals compared to wild animals associated with the other people who were living in Europe at the time, which I'm going to be referring to, the hunter-gatherers. There's also quite a lot of studies done on the human remains, on the skeletons, about what type of diet they had. So you can see that in isotopes of carbon and nitrogen, and figure out what type of diet they've had. And it's, again, clear that they've had a different diet, the farmers and the hunter-gatherers. There's also some very iconic differences between the ways of burying their dead. So typically the hunter-gatherer grave is in a cave or is in a flat bed straight down into the ground, whereas at least many of the sites associated with farming have these megalithic monuments as graves. So here's one from the western part of Sweden. There's a lot of them across the European continent in different forms, but very large constructions in stone. This question about the potential of the spread of agriculture associated with this Neolithic new package has drawn a lot of attention to scientists for quite a while. 100 years ago, there was quite a lot of arguments among archaeologists of how that spread could have happened. And if we jump into the more recent terms, or recent times in the 60s and 70s and 80s, Luca Cavalli-Sforza and colleagues started using genetics from modern day humans to try to get some sort of glimpse of patterns across the European continent. And what he found was basically a gradient in principal component 1 based on a handful of markers, typically blood groups and HLA markers across people in Europe. And he found this gradient from the southeast towards the northwest. And with this gradient, he argued that there was a population replacement due to the agricultural revolution. However, at the same time, most people in archaeology was arguing a different theory. They were arguing that it was the same group of people living across the European continent, and it was only the culture or the new way of life that was spreading. So it was a spread of people or instead of migration. To complicate the matter a little bit, we can look into patterns of variation across Europeans today. And a lot of people have done that and try to see if there's anything that can be used from these patterns of people today to say something about these early migrations, or if there was any early migrations. So if you stare at this relatively famous principal component analysis of some 1,300 individuals, humans from across Europe, you see that there's a pretty good correlation between genetic similarity, and you should view each little dot here, or little letter code, as an individual, and closeness on this PCA means that they're genetically similar. So you see similarities among people from Portugal, Spain, France, and then you see gradients and as you travel around this genetic space, and you see that it's pretty well correlated with geography. So in one way, it's rather easy to understand the current genetic variation across the European continent as an isolation by distance model. That doesn't fit very well with what Luca Cavalli-Sforza was proposing, a range expansion or a replacement from the southeast to the northwest. However, if you dig even deeper into these modern day patterns of variation, you do see gradients or correlations with geography that goes from south to north. So the strongest correlation across Europe, correlation between genetic variants and geography, is from the south to the north. So that might be an indication that there is something going on in that particular axis. But if you start modeling that in a proper way, based on standard population genetic theory, and you send a range expansion from one area, and you basically replace the people who were living there before, so you have no admixture, then in fact you should see the greatest gradient in PCA or in some correlation should be perpendicular to the range from where the migration came. So in other words, these correlations that we do see in the European continent doesn't really fit well with a range expansion or a replacement of people from the southeast to the northwest. So to solve this problem, people started, or researchers started, turning to studying the people who were living in the times that you're interested in. So instead of trying to figure out past events by looking at the variation that we see today, we started examining the DNA in the skeletons of the people who were living 5, 6, 7, 8, 9, and 10,000 years into the past. And I'll just turn to Scandinavia now, because it's one of the places where it actually first started emerging, this pattern that I'm going to be explaining. So in Scandinavia, we're kind of fortunate in that it's cold. So that means DNA preserves pretty well, especially in past remains. And there's also not a huge agricultural activity across the entire landscape. So there are reasonable amounts of remains that you can dig out from the Stone Age. There also happens to be coexistence of these two groups. So up until this red boundary in Scandinavia, you have the expansion of farmers or the farming culture until maybe 5,000 years ago. At the same time, there's another group or another culture existing around these coasts and on the islands out here in these blue areas of modern day Sweden. So in Scandinavia, we have a unique opportunity that both of these two groups actually coexist at the same time, whereas in more central and southern Europe, there's a very rapid shift in the archaeological record from a hunting and gathering way of life to a farming way of life. So it's unclear whether there's been any overlap in other parts of Europe. So one of the first studies that was done on multiple ancient individuals from these two groups was from remains that are about 5,000 years old in Scandinavia from these two different cultural groups. And to many people's surprise, one of the most striking results was that these two groups, the farmers and hunter-gatherers, they're genetically very distinct. So if you remember the plot that I had from the November study, we basically have the same representation of modern day Europeans and people outside of Europe in these gray dots. And then if you stare at the colorful dots, all the red ones are from sites that have been associated with farming, and all the blue ones or beige ones are from hunter-gatherer sites. So the first observation is that, well, they're really two distinct groups. So the farmers or the individuals from farming context, they're genetically more similar to people who today live in the Mediterranean areas, whereas the individuals from hunting and gathering contexts, they're genetically outside the range of modern day Europeans. And if there's any group or any direction in this two-dimensional geographic representation of genetics in modern day Europeans, and it's towards the north and northeast of Europe. So that's showed what had been argued for a very long time between archaeologists on the one hand and geneticists maybe on the other hand, that it's really two distinct human populations that practice two different ways of life. In other words, the farmers are newcomers, they're migrants over probably hundreds or at least up to 100 generations, so it takes quite a while, but it's a new group of people coming in to Europe from the southeast. So let's zoom out from Scandinavia and let's see if we can see any general patterns. So since the early 2000s, maybe 100 or more than 100 studies have been done on Neolithic remains from Europe and now from other parts of the world. And here's some summaries of a number of those studies a little while ago. So again, we have these colorful figures from modern day Europeans. We basically see the same patterns. We have the south, we have the north, the northeast maybe, and then east and west pretty much on this representation of genetic variation today. So if we jump back to prior to the Neolithic Revolution, we should focus on these individuals. These are the younger ones. We have kind of a range of hunter-gatherers that are genetically outside the range of modern day Europeans. These are gray ones. This would be the northeast or the east, and this would be the southwest individuals from current day Iberian Peninsula. So we see the same patterns as we did in Scandinavia. Hunter-gatherers are outside the range of modern day Europeans. But if we jump into the Neolithic time period and start looking at individuals who are associated with farming, then they are all genetically very similar, irrespective of where they were excavated. So we have excavations from Hungary, Anatolia, Central Europe, Iberia, and they're genetically all very similar. And now we're in the time frame of maybe 8,000 to 4,500 years into the past. There are some individuals who live in this time period too, associated with hunting and gathering sites, and they're genetically similar to the other hunter-gatherers or the previous hunter-gatherers. Some of these cultures live on longer, for example, in Scandinavia. Interestingly enough, if we move forward in time, we move into the Bronze Age, and now there's more additional gene flow coming from the east, and I'm not going to go into the details of that. But if you start comparing the genetic makeup of people in this time period from Central Europe, you're starting to see a correlation with geography. So you have individuals from the east, you have individuals from Central Europe, you have individuals from southeast of Europe, and you're starting to see the same patterns as we have today. So in other words, during the Stone Age, the genetic makeup seems to be much more strongly correlated with a particular group that live in a specific way, whereas in the Bronze Age, we're starting to reach this correlation with geography.
Matthias, you have 5 minutes left.
Okay, 5 minutes. Okay, I will speed up. So how different are these groups? Well, we can compare modern day different groups with using FSTs, for example, and then we can compare the hunter-gatherers, and it's quite clear that they're more distinct in the Stone Age than people are today. Another observation is that the diversity in the farmer groups is greater than it is in hunter-gatherer groups. And that we can understand due to larger census sizes in the farming groups and then larger carrying capacities in that particular way of life. I'll skip that slide and then we'll go very quickly here. So another question that arose here was, where did the farmers come from? And the solution to that is to dig into various parts of Anatolia and the Middle East and check the people who were living 10,000 or 6,000 years ago in these areas. And the first observation that you have there is that the people who live there today are not direct descendants of the Neolithic groups that were living there 5,000-10,000 years in the past. The greatest genetic similarity is actually among people of Southern Europe today. But if we compare the individuals from the Neolithic Anatolia, for example, Barsin, Bonjuklu, Tepchik, they're similar to the Neolithic Europeans. And that links that migration back to the Middle East and back to Anatolia. Let's just end with a question of who migrated. We can investigate different migration patterns and different admixture patterns. And we're now focusing on the migration from Anatolia, and I'm just going to compare it to a later Bronze Age migration. And what we can observe in the migrant or the mixed groups is that during the Bronze Age and the migrations from the East, we have way too few X chromosomes in the mixed individuals, whereas during the Neolithic expansion, we have about the same X to autosome expected ratio. In other words, it's whole families, it's whole groups that are moving during the Neolithic expansion, whereas it's predominant males that are moving during the expansions from during the Bronze Age. Last thing, comparing to patterns of plants. So I was involved in a study quite a long time ago with Arabidopsis, and we had pretty much similar types of questions about range expansions. And it turns out that the most likely expansion of Arabidopsis into Europe is in the last couple of thousand years, and it coincides with the migrations of people into this area. And the gradient is really going from the west to east. So if you remember Olivier Francois' work earlier, this really shows a possibility of a complete replacement, or more likely a new expansion into a new area from the southeast to the northwest, probably associated with agriculture disturbing the land, and Arabidopsis really loves disturbed land. It's a weed. So with that, I'll say that we have an ancient DNA facility here in Uppsala that's exploring the possibilities of getting DNA from ancient plant remains. So Concetta is one of the researchers driving that here at Uppsala, and some of you might know her from Montpellier. So we hope to be able to get DNA from ancient plant material from archaeological excavations. And with that, I'll end. I'm happy to answer any questions that you might have.
Thank you very much, Matthias, for this very insightful talk. It's quite amazing to see how much we can tell about our own history from old bones. So we will switch to a short session of questions. We will start with questions from the room, also for methodological problems, because there's a slight delay with YouTube. So if someone has a question, we have a microphone that we can give you. Take the opportunity of being there. So maybe while we are waiting for some questions, I will ask a quick one myself. Matthias, I was wondering whether the archaeological data and the ancient DNA can tell us something about sedentarity. I mean, whether we can tell whether it happens before the spread of agriculture or after.
Right. You mean if we can tell whether certain patterns or processes happened before or after a certain time period, or if people were sedentary?
I mean, if the genetic data can tell us whether there is, for example, less migration, less movement before the spread of agriculture.
Okay. Well, I think the best tool we have there is strontium isotopes. So strontium isotopes can tell you whether so strontium isotopes are quite well associated with the groundwater and bedrock makeup. So you can figure out whether an individual, if you're looking at humans, for example, have been living in the same area for a very long time. And you can do that by taking different teeth, for example. So if you take teeth that you get when you're 6, 7, 8 years old, then you get a pretty good view on where that person was living at that age. And then if you look at the skeleton, for example, the skeleton is basically replaced every 5 years. So it's an account of where you were living late in your life. And then you can see if a person has been very sedentary. I think there's pretty ample evidence already about that hunter-gatherers, for example, they were much more mobile, whether they're like large-scale migrants or migrating very large areas, or if they're just moving around in a large area. That's hotly debated in the archaeological community, I think.
Thank you. So there is a question in the room.
Okay, so thank you so much, Matthias. And may I ask if there is information on the relative role of cultural versus migration mechanisms in other Neolithic revolutions around the world, in America or the Far East or wherever?
Right. That's a very good question, and I know a lot of people are trying to see if the same type of patterns appear in, for example, Americas. And this far, I haven't seen anything that contradicts the same processes that we see in Western Eurasia. But at the same time, I don't think there's been any studies that really conclusively shows that that the same process is happening in the Americas, for example. The same should probably be possible to do in South Asia and East Asia. But then you also have the complication of that there is some connection to the western parts of Europe, and we know that there are migrations that are going also eastward. So maybe island Southeast Asia is the best place to, or another place to do that. And I know there are indications of expansions connected to agriculture from New Guinea, for example, and that might have been a spread of people also. But I think that's something that we will hopefully learn over the next couple of years, where more ancient DNA studies are done in other parts of the world, in particular connected to Neolithic expansions.
Hi, I'm here. I'm Jacques David. So I have a question on, you said that the effective size of the farmers was greater than the hunters. Did I understood well?
Yes.
Okay, so does it mean that you can have any evidence of the, I would say, a more efficient selection for adaptation to any diet or concerning domesticated plants or animals, or any sign of adaptation to new climates? So have you ever looked at that, or do you think that you have sufficient power to detect if man, since it has become farmers, because of these large effective sizes, the new mutants that are able to come into the populations and the efficiency of selection, could you see whether it was easier for farmers to adapt compared to hunters?
Right. So I mean, that's a very interesting thought also. And in principle, it should be, I mean, selection should be more efficient in larger population sizes. We know that from population genetic theory. I think personally that time is a bit too short. So there hasn't been enough generations to actually get to these adaptations, or at least not in a larger sense. So there has been studies on, for example, amylase production in farmer communities or versus hunter-gatherer communities. And there are some initial results that point to that people living in farming communities have more copies of the amylase genes. But there are also other studies that say that initial study didn't actually cover a large range or a large number of population, and that it's not as clear, those types of patterns of larger or more copies of the amylase gene, for example. Lactase is another classic example. In that case, I think the selection is so strong, so it would probably work irrespective of the effective population size, whether you had that pattern in a smaller effective population size population, that would still hold. So I haven't seen any general results that point to that selection should be more efficient in the farmer communities, simply because I don't think there is enough time since these two ways of life distinguish themselves.
We have time for one last question. May I ask one if, so you showed at the end of your talk that you could use plant data to test for alternative hypotheses on human migration. Could you use crops instead of wild plant species to test your hypothesis?
Yes, I think a lot of people are interested in doing that and following different, maybe not, you probably want to avoid the heavily bred crops. You want to use the land races or these types of crops. And I know there's interest in that in Scandinavia, for example, and in other places. So I think that's a very interesting avenue for getting more details into how this Neolithic revolution happened across Europe and in other places.
Well, thanks a lot, Matthias. And I think we have to move to the next speaker.
Thank you.
Thanks, Matthias. So after this focus on human, we will switch to crops. And we are welcoming Hernan Burbano from the University College London, also online, who will present some of his research to understand how crops have adapted to a large set of environments during the expansion after domestication. Hernan, you can start sharing your screen and speak.
Yeah.
Perfect, we can hear you.
Okay, perfect.
Thank you.
All right, so good morning, everybody. First, I would like to get feedback that you are listening me correctly, and then you are seeing my slides, not in presentation mode, but like in presenter view.
It's on the present. You can put it full screen, maybe? Because now we see your next slide.
Now?
Perfect.
Okay, very good. Okay, again, good morning, and it's a pleasure to be here giving this presentation. And today I'm going to link some of the topics that Matthias already started to discuss about. And the title of my talk today is a retrospective view on plant genetic diversity using ancient DNA. And what is this retrospective view about? So this retrospective view consists in retrieving DNA information from the past. And I'm presenting here in this slide how this arrow of time can inform evolutionary analysis if we can retrieve DNA from the samples as the ones that I am presenting here. For example, sediments, ancient animal remains, such as the Neanderthal bones, as Matthias presented before. But I'm going to focus on plants. So from plants, we have two main sources. We have plant archaeological remains that can get us back to thousands of years before the present, and also herbarium specimens, which would allow us to investigate processes that happen in the order of magnitude of hundreds of years. In addition to looking at plants, one can also, in some instances, look at plant pathogens, since some of these plants present lesions that are compatible with past outbreak of plant diseases. And in addition to these microbial pathogens, one can also, using the same approach, start to look at other actors that interact with plants. For example, insects, if we can retrieve DNA from museum specimens. So in my laboratory at University College of London, we use all the samples that I present here to study like the evolutionary history and adaptive history of plants and plant pathogens and pests. So, but how does the DNA that are present in the sample look like? So this is what we call ancient DNA, and the DNA in a nutshell is very degraded. Here I'm just showing how the length distribution of the reads sequences from those samples is very short, about 50 base pairs. One needs to understand that this is a metagenomic type of approach, since not all the DNA is endogenous. That means that it doesn't belong to the host, but we have a lot of microbial DNA present there, some of which we're very interested in. And in addition to that, when we sequence the DNA, we encounter a series of nucleotide misincorporations. Here I'm simply showing the percentage of different misincorporations along a DNA molecule, and you can see this excess of C to T substitutions. So this is to show you that sequencing ancient DNA and analyzing it is slightly different from modern DNA, and we need to take these characteristics into account while we extract the DNA, when we prepare the libraries, and when we analyze it bioinformatically. Today, I'm not going to get into detail about the methods, but I'm going to highlight that it's very important to generate ancient DNA data sets of very high quality. And I'm going to do a little shout out to this paper that we published together with my former postdoc, Rafael Gutaker, where we present very comprehensive protocol that include the isolation, the library preparation, and also the basic bioinformatic analysis of DNA retrieved from historical and ancient plant specimens. So after this little technical introduction about ancient DNA and what I call the retrospective approach, I'm going to move today and talk about plant domestication. But in particular, I'm going to talk about the spread of agriculture. As you can see in this beautiful map, you have different crops, and they are located in a map where they originate. And you can see how different regions of the different regions of the world, different crops have been domesticated. I would like to point out that some of them were domesticated around the equator of subtropical areas, and only post-domestication, they move across latitudinal gradients all around the world, for example, to the temperate zones. And this is the process, the post-domestication process that I'm very interested in. So one of the questions that we have asked in my lab is, how did crops spread and adapt outside of their native range? And we're talking about this post-domestication process. So for that, I'm going to present today two snapshots, two case examples of different crops moving across latitudinal gradients and how we studied them using a combination of present day and ancient in the first case and historical samples in the second one. So the first case today is about maize, and it's about ancient maize adaptation to temperate North America. This is a work that we published a couple of years ago that was led by Kelly Swartz, now at the GMI in Vienna, and Rafael Gutaker, now at Kew Gardens, in collaboration with the very well-known maize geneticist Ed Buckler at Cornell. And we're going to look at flowering time in maize. And why we look at flowering time? Because we're going to use flowering time as a proxy for temperate adaptation. And what is it so? So if we look nowadays at modern breeding lines of maize in North America, you can see that flowering time is stratified by latitude. You see that the breeding lines here in blue from the north need to flower before that's the one in the middle USA and before the ones in the south of the USA. And the logic is that if you are really up north, up to the border with Canada, if you don't flower as a maize plant early, then winter is going to come. So some important facts about flowering time is that is what we call a complex trait, which means that it's many loci with very small effects all along the genome, but it has a very high heritability. So this is something comparable for human genetics is also in the room with human height, which is also very complex, but with a very high heritability. If you see here the map of the Americas that I'm presenting, this is color coded in this white till red palette. And this is colored based on what is called growing degree days, which is a measure of heat accumulation. So you can see that is more to the red in the tropic, but when you move to the temperate zones, we have less heat accumulation and the colors get more pale. So what about maize domestication and the introduction to the Southwest? So we know that maize was domesticated about 9,000 years ago in the Balsas Mali in present-day Mexico. And then it started to move around the continent. So it reached, it moves northwards, and it reached the Southwest about 4,000 years ago. And when it reached the Southwest, agriculture started immediately there. But although it also reached the temperate zones 4,000 years ago, agriculture started there 2,000 years afterwards. And you can see the latitudinal movement was not as big as moving from Mexico to the Southwest. You see the changing in the color palette in the map, which is really getting in the temperate zones when it's going to experience frost already. So we know from archaeological evidence that agriculture was delaying the temperate uplands for about 2,000 years. So we wanted to understand that process, and we're going to understand this process using flowering time as a proxy for this temperate adaptation. Fortunately, we have samples from archaeological remains in the Colorado Plateau in present-day Utah, and the shelter is called the turkey pen shelter. So from now on, I'm going to talk about turkey pen shelter maize. This is a view of the stratigraphy of the column, and you see on the right-hand side numbers, and these numbers correspond with the maize cups that are shown at the bottom. So those are the type of samples that we're working with. So the samples were C14 dated to be about 2,000 years before present. So exactly the time when agriculture was starting in the temperate lands. And we took a shotgun approach to sequence those samples. So I'm not going to present details about the sequencing, but we had the fortune due to very good preservation in a very dry environment to be able to sequence whole genomes from, I think, about 16 of these maize cobs. And what we wanted to do is perform phenotypic predictions. And I bring this picture because this is the kind of headline that people normally read in newspapers. The headline could be Neanderthals have red hair or have blue eyes or any other characteristic. And this is a very simple phenotypic prediction because you will go and look at the SNP at a particular gene that is known, look which variant is present, and then infer what is the phenotype. So the challenge that we have was very big because what we have were these maize cobs from which the genomes were sequenced, and we wanted to know at what time they were flowering 2,000 years before present. So in the next part of the talk, I'm going to show you the methods and the approach that we use to carry out these phenotypic predictions. So the first thing to know is that to do that, we resort on maize land races. And maize land races are locally adapted cultivars. And this is a distribution of the land races that we use in the study in the America. If you look at the upper part of North America, you see this pink star, that is turkey pen. So when we summarize the genetic relationships of these samples using a dimensionality reduction method, such as MDS, very similar to the PCAs that you saw in the previous presentation, this is how the land races look like. The color code is the same as in the map. And I want just to point out to the pink samples, which are the turkey pen samples. And the important thing is that the genomes from these samples 2,000 years before present is not like outside the variation of present days maize land races, but it was from within the same variation. So it is part of the variation. This is going to be important for the phenotypic predictions. So to carry out the predictions, I'm going to show you which data sets and which roles they play. So first, we use the inbred lines of North America that I already introduced to you at the beginning. And we know that they have flowering time variation, which is stratified by latitude. So that was the training data set that was genotype and phenotype before we start the study. And what we did is that we sequenced the land races. They were genotyped, and also they were phenotyped for this study. So those land races were grown over 2 seasons in 3 different states in North America, in Arizona, North Carolina, and New York, and they were phenotyped for flowering time. So the method that we use is a method that is highly used in plant breeding, that is called genomic selection. And what you do is try to predict the phenotype based only in the genetic relationships. So what we could do in this case is that we can mask the phenotype, train the model, uses only the inbred lines, and then predict the phenotype of the land races. And we can ask how well were we doing. So we were predicting with an ability of about 70%. So this is the correlation of the predictions they observed from the land races. And then the jump that I will ask you to do is that if turkey pen variation, the archaeological samples, is within the variation of the land races, and if we can predict with a 70% ability flowering time of the land races, then we have a pseudo-validated way to carry out the prediction on the turkey pen samples. So here I'm going to show you how the predictions look like. So here you have a way to phenotype flowering time. These are days to antithesis. You have in the y-axis the day to flowering, the days to antithesis, and then you have the different samples there, but I walk you through. So here in this red square, you see the tropical samples that they can flower late. And then we move to the temperate samples. We predict, of course, that they need to flower earlier. Then the question is, where is turkey pen there? So what we found is that turkey pen is already within the variation of temperate land races. So which means that was already partially at least adapted to temperate. That's what agriculture took off at the time. So for this, we can conclude that using this type of methods, we can predict previously unknowable complex traits in archaeological samples. And in this particular case, turkey pen maize was early flowering and marginally adapted to its environment. The last part that I want to present about maize is that we ask, where was this adaptation coming from? So what we did is that we look at genome-wide SNPs and ask how frequent those SNPs were segregated in maize wild relative teosint. And you see that about 82% of the SNPs are segregating in teosint. Now we look at the SNPs that we think are involved in the adaptations, and we ask how frequent they were found segregated in the wild relative teosint, and we found that they were about like 90%. So from this analysis and other, we conclude that probably this adaptation that was selected for flowering, for early flowering, was present also in standing variation in teosint.
Bernard, you have 5 minutes left.
Okay, and so alleles for temporary adaptation were selected from pre-existing variation. And then the last part is a very similar question, but it's looking at potato. And this is the adaptation of potato to Europe. This was done also by Rafael Gutaker. So very short, potato have the same kind of problem, was domesticated in the Andes. In the Andes, only form tubers during short days, but not over long days, which is not a problem in the Andes because you have 12 hours of daylight throughout the year, but they moved to Europe very recently, where short days are going to come close to winter. Interestingly, potato was found moved in prehistoric times also to southern Chile, where it can tuber on long days. In contrast to maize, the genetic architecture is much simpler, and this gene, CDF1, has allelic variation that determines tuberization. So the 1.1 variant doesn't permit the tuberization on the long days, but we have two additional variants. We have inactivation that permit the tuberization, as shown here, the wild type not tuberizing, but if you overexpress, for example, the 1.2 variant, it's going to tuberize on the long days. The approach that we took was similar. We took our variant samples starting from the 17th century, such as the samples here. Some of them collected by Charles Darwin when she was on the Beagle. And then we combined that with present day diversity. So a couple of glimpses through the changes in Europe. You see this is a phylogenetic tree of the chloroplast genome, and you see how the chloroplast genome through time, it segregates between Andean and Chilean land races and also wild potatoes. You see that it changed from from Andean to Chilean through time. When we look at the nuclear genome using a statistic that is called F4 ratio, you see that the nuclear genome recapitulates what we find in the chloroplast, that we find this decrease of Andean ancestry through time. So the potatoes collected during the middle 17th, 18th century were related to Andean land races. And after their introduction to Europe, they had mixed with Chilean genotypes. Then we look at the allelic diversity of CDF gene. And in the picture that I'm going to show this type of donuts at the bottom that is going to show you the state of CDF1. If you see at the outer part, and it is gray, is the active variant, the one that doesn't tuberize under long days. And if you see any color, is that it is tuberizing. And in the middle, you just have a geometrical figure showing the type of chloroplast. This is how it looks in the Americas. I walk you through the figure. You see that in the Americas, we found only gray. So that was only the active variant that doesn't tuberize in the long days. So the inactive variants were not segregating in the Andes. So they're probably not coming from a standing variation in the Andes. When we look at Chile, it was very interesting. Unfortunately, the 20th century samples were not mixed. But the samples collected by Darwin in the 19th century, we also found only the active variant. So we said that the present day Chilean land races are mixed with European cultivars, but only the active CDF variant was present in 19th century historical Chilean sample, which means that those potatoes in Chile are adapted to temperate regions on a CDF1 independent way, which is really exciting. But we asked the question about what happened in Europe. So we look at Europe, you see that at the beginning, here it's shown with the red squares, you have only the active variants, but then through time, you have the appearance of the inactive variant that allow tuberization and adaptation. So the adaptive variants were detected in Europe during the 19th century. And we think, based on the data that we have, that this variation may have appeared de novo in Europe. So with that, I like to finish. And I talked today about the spread of agriculture across latitudinal gradients. In the first case, we look at flowering time. We have a very complex genetic architecture. For that, we use archaeological samples to go to prehistoric times. And we found that adaptation comes from a standard variation, and adapting maize took early agriculturalists about 2,000 years. In potato, we look at tuberization onset time, which have a much simpler genetic architecture. For that, we have a different time scale. Using herbarium specimens, we went to historical times, a couple of centuries before present. And in this case, we found the adaptation come from the novel variations, and we look at centuries. So with that, I would like to finish. Thank you again for the invitation. I'm going to thank the people involved in these studies. I already mentioned Rafael and Kelly that did most of the work. And I'm looking for people to join my lab. If you are interested in this kind of topics, please reach out for writing postdoctoral proposals. And I'm very soon offering PhD studentships too. And then I would like to thank my institution, the University College of London, and also my funding sources. So I'll be very happy to take questions.
Thank you, Hernan. Do we have any questions in the room or online?
Yes, about flowering time for maize. How was measured this flowering time? Because we know that if you take a maize from the tropical area, you put it simply in a temperate area, the flowering time will be very different. So how did they measure this stable flowering time figure?
So this is how, so the question is about how were the land races phenotyped?
Yes. How could we compare the flowering time of a tropical and temperate maize, where they are measured in a tropical area with a short day length, which is very different, or where they are measured all together in the temperate area?
Okay, yeah, thank you. Yeah, I understand. So yeah, exactly. So yeah, to have like kind of environmental variation of the reaction of these different land races. So this was part of the work done by my collaborators in Cornell. So what they did is that they grew the land races in 3 different locations across latitudinal gradients. So they grew up them in over 2 seasons in Arizona and over 2 seasons in North Carolina, and then in New York. So you see that we have this latitudinal gradient already, and then the same land races were like grown in these 3 different parts. So that would take into account a bit the environmental variation. So the comparisons is among land races that are grown in the same location. And then the prediction is based on the land races.
Hernan, I had a slightly technical question also. I was quite amazed by the prediction of the flowering time on the archaeological sample, the Takipan. There's very low variation in the predicted flowering time. Was it because there is little variation, genetic diversity in this sample, or and eventually did you try to predict other traits?
Yeah, so thank you for the question. That's an excellent question. Yeah, so there is like, so also we don't have like a lot of samples. So we have like the population of turkey pen, we have like 16 samples. So we don't know to which extent are we sampling the genetic variation that was present there. But when we compare the variation with other like land races, it doesn't seem that it's like extremely bottlenecked population. So the pairways like genetic distances between them is not so reduced, but then you're right, like the variation was very narrow. The second question is about predicting other traits. So we did. So for example, if you look at the paper, we predict also height, which have a very similar genetic architecture, and the prediction ability and accuracy of height were very similar to flowering time. And then, for example, we try to predict like the tailoring, so how compact the plant would grow, how narrow or how broad. And in this case, the predictions were like really bad. And the reason is that you are going to make your training data set using modern breeding lines, where they are grown like very narrow. You don't have also like phenotypic variation, and therefore probably genetic variation for that. So one of the things that is important, you need to have training data sets that represent genetically and phenotypically what you are going to predict. So for height, the prediction was very good. For flowering time was good. From tillering, it wasn't. And of course you can, I was kind of making a joke with the Neanderthal slide, but of course you can look also like a monogenic traits. So you can look if, for example, it was accumulating beta carotene or not, things like that. And those are predictions that also we did, and it seems that that was not the case.
Thanks a lot, Hernan. I think that we have to move on to the next speaker. So Yves, who is in the room?
So Yves is the exception part of Arcad, but is going to present some of his research, maybe digging a little bit further into the evolutionary dynamics of crops, using research he's been carried out for some time already on pearl millet.
Okay, thank you for inviting me to do a speech in this special occasion. So I will speak about the history of domestication and how it's helped us understand how crop dynamics occur during the last 10,000 years. So the reason why we are interested in the study of this crop evolution is also because there is it's at the same time the study of the population that cultivated them and how the society and agricultural society emerged and developed. So domestication process is a process where we have at the beginning some wild plants, and by human selection and adaptation to different environment, we have a development of morphology that is suited for agriculture. So in cereals, it's been larger spike with a lot of seeds and a more compact inflorescence with less ramifications. So here we have the example for permeate, which is a cereal from Africa. So first I will go through an example and a study we done in African rice. So the question we will ask in this species is how the diversity is in the cultivated compared to the diversity of the closest wild relatives. What were the dynamics in terms of diversity or effective size in the crop during the expansion of agriculture, and if we could trace back some origin of the crop, if any. So the first thing we do for this crop was fully sequence land races and wide and cultivated plants. So you have here a PCA, and in green we have the cultivated, and somehow in the PCA they are in the corner, like showing a lot less combination of alleles than the wild species, which cover almost all the graphs. So what happened, and it's happened pretty much all the crop, we have a diversity in the cultivate that is a subsample of the wild. And so maybe because the origin of the cultivate occurred in a small area, or because the cultivate were with a very short effective size during a long time. So diversity generally in cultivated species are much lower than the wild relatives. Then we could try to understand what will be the dynamics. And we have now a very powerful tool using population genetics that help us understand the size of the population and its evolution through times. And it's come back to a theory of how two alleles relate to each other and derive from one another, and coalescence theory. And we have a direct link between the type of the population and the time when the two alleles coalesce. So there is a several method that will develop based on this simple principle. So if we have a size that is large, then allele will coalesce in a longer time periods. But if for some reason in the past the population was smaller, then we don't have the coalescence we expect based on large population size, but we have a new coalescence time that is shorter. So if we analyze the phylogeny of the alleles, then we have an inference of the effective size during the past, for example, 10,000 years in crops. So this kind of method lead to this kind of graphs. So you have the time at the x-axis, and you have the population effective size, and this is estimated for the cultivated rice from Africa. So rice in Africa is a different species from the rice in Asia. And what we observed is around 6,000 years ago, some kind of decrease of population size, then some kind of little bit low population size during almost 3,000 years. And then beginning around 3,000 years, then we saw a strong increase up to 1,000 years, and then recently a small decrease. So how do we interpret this graph based on the knowledge we have and the archaeological knowledge you have? So we know that we have archaeological remains that tell us that rice was domesticated at 3,500 years ago. So during this period there, certainly the domestication happened. And then we have certainly here a strong expansion of agriculture. And so effective size of rice began to explode. And then in the last 3,000 years, we have some kind of recent crash that we know might be linked to the use of others crop in Africa, and African rice was replaced by Asian rice in some place, for example. So this could be done for the whole sample, but some of the history we have here might be linked to the history of the wild relatives. In this graph, in the past, at some point, we moved from the cultivate to the wild. So when and how could we assess the difference between the cultivate and the wild? So we use a similar methods, but this on individuals. And here we do the analysis for the wild, and they are in purple, or with Labarthe, and the analysis for the green, the cultivated. And so what we saw is that the decrease we observe at 6,000 years ago is not specific of the cultivated. It's a pattern that was occurring also in the wild population. And we have a difference between the wild and the cultivated only after 3,500 years ago, which corresponds to the period where we find the first archaeological remain of cultivated rice. So by comparing these two patterns, we could say which is different from the wild and which is common to the cultivated, and which is very specific to the history of the cultivated. So here, one thing you have to retain, because I will come back to it, this strong decrease that is not really specific to the cultivated. And so could we then move to the geographic origins? So now we have tools, and we use some tools with Olivier François in Grenoble to infer the inference in terms of probability of cultivated rice and the origin of cultivated rice, where the expansion of African rice agriculture began. And so this uses statistical models, but the result is pretty simple. It's kind of heat map of probability. And we have a red zone around the delta interior of the Niger River in northern Mali. That is a hotspot where we think the origin of the expansion of rice agriculture occur. And you have a dark point there, and that's the oldest archaeological remain of cultivated rice we know around Mopti, a town named Mopti. Yes, so, okay. So we get this perspective using one crop, but what's might be interesting and maybe interesting in linking this research on cereal to study of evolution of population and agriculture is also to look at it from a different crop perspective. So we did similar analysis for the cereal, permeates, yams, and also more recently fonio. So what the result of this different analysis tell us? So here you have the inference we showed previously for rice. So the analysis of permeate give a slightly upper latitude, but in the similar regions, and the analysis on yam give a southern attitude, but always on the kind of Niger River interior delta or south of the Niger River delta. But results from fonio and preliminary result from fonio tell a different story about the origin. We don't only have one big spot of origin in West Africa, but we trace the origin of fonio based on chloroplast diversity, but also based on the same methodology we use for the other crop in a more eastern part, more Central Africa eastern part, north of Nigeria. So this kind of analysis using several crops help us understand that the dynamic is quite complex. We might have some area where some crops certainly emerge one after another, and other area where other crops emerge differently. And all these crops nowadays are cultivated throughout West Africa. So one question that is an old question we have in studying domestication is, okay, what was driving this process? So it's a difficult question, but we might have some hints about an answer, and for that we will come back to domestication of permeate. So I told you that we made inference about the origins, and one the big surprise of this study when it popped up on the computer was that the origin was actually in the Sahara. So it's a little bit difficult for us at the beginning because it's a desert. So why the origin was so north? And so to understand a little bit this, we also have here the position today of the wild relatives that are the closest to the cultivated, and they are this blue dot around this blue area, so they are further south. And the question is, was this wild relative northern some 1,000 years ago? So one thing that happened in the Sahara is that we were on the green Sahara several thousand years ago, and we moved to the actual Sahara we know today and settled this dry area around 3,000 years ago. And so we have here some example of pollen collect inside some lakes that are found in the east part of the Sahara. And when we look at the different type of pollen, the green one are the tropical plants pollen, and we saw that they slightly disappear, and they disappear around 5,000 years ago. And what appear is more acacia, which is more crop that plant that are adapted to drier environments. So we know that the patterns of evolution of the Sahara were pretty wet during the 6,000 years ago, and begin to dry fast after that. So we then estimated. So we combine our genetic data and with the archaeological data we have for occurrence of cultivate permeates, and we end up with an estimation of when the diffusion of permeate begin, and we end up with the time that is 5,000 years ago. So one kind of hypothesis we could make is certainly, and we certainly have the same thing from the previous analysis on rice, we show in the wild species that the effective size decrease. So it's kind of a signature of less good condition for them, okay? And so certainly with the evolution in the Sahara, we have less suitable environment. And we saw it through wild populations, and certainly less also, and it's then have consequence on population that were at this period, and maybe that was one of the driver of the change and the adoption and the development of farming in Africa. So it's difficult to draw a very conclusion from that. They are just like co-occurring phenomena, but I think it's important to have this kind of idea today when we have major climatic change, and in the past it seems to have been linked to major social change, which is the adoption of agriculture. So the perspective we are here is major climatic change in the past, drying of the Sahara, major change in society, and maybe adoption of agriculture as a way to move forward. So I have other small key message from the study we did in the last 5 years. One point we found is that inference about domestication and diffusion of plant is always more complex than you think first. And we have, for example, a lot of different paths of diffusion, even to the same regions. So this present an analysis that is done on chickpea, the work we collaborate with. And so the origin of the domestication go back to the Fertile Crescent, where we have the plants that are kind of the closest to the wild relatives. But when we try to make inference about path of diffusion, when it's the story is pretty complex. We have two diffusion to the east, one by the the Black Sea, and one through the up to India. And then the samples that are in Africa are certainly derived from the first wave of migration and to the south, and the migration to the north is associated to the diffusion to a Mediterranean area. So scenario of diffusion of plant might be quite complex, and that's one thing we learned along the way of the different study we did. The other important thing that is emerging across a lot of studies is that we have a lot of hybridization during the diffusion of crops. And this diffusion from the origin of the crop then meet new population, new wild population. And this led to new diversity, bring back to the cultivated, but also it sometimes helps population to adapt to new environments. So one example we study is the diffusion of permeates. So we have an origin in West Africa, but then permeates migrate to the west, up to Senegal, and to the east, up to South Africa and to India. But what we observed when we study the diversity of this crop is that we have two major hotspots of diversity, of rare allele diversity, one in the east and one in the west. And we could demonstrate that they are linked to gene flow between locally occurring wild relatives and the cultivated. So after the origin, after the diffusion, a lot of hybridization event occurs, and crop then ruptures the load in terms of diversity. And this lead, we could prove in some case, to new adaptation. And of course, this story of plant are embedded in human population history. And one of the recent results we show was the co-variation of the structure of population, so how diversity is organized geographically, with cultural traits, in this case language. So we have diversity of the cultivated plants that co-vary with a cultural trait, which is the language of the people that found them. So I have so many people to thank. I didn't begin to write their name. It was too much. I couldn't have only one slide on this. So large part of this work was funded under the Arcad project and Agropolis Foundation, but also INR, French funding agency. And we have a lot of collaboration that were between the IRD and CIRAD for all the work I presented here, and other partner in southern country. So I thank them all. I don't have a lot of room to put them in one slide, but it was a lot of work between different researchers. Thank you very much. If there is any question?
Thank you, Yves, for your talk. I have a question. I'm really impressed by the crop effective size over time. and the fact that it decreased by 3,000 years ago. And you relate this tree to the climate factor, isn't it?
For the three, so the 6,000 year decrease or the recent decrease? The recent decrease?
Yeah.
The very recent decrease. So among the hypotheses we made is not about climate. The hypothesis we made is about arrival in Africa of a lot of different crops. Today, African rice is largely replaced with Asian rice. We know also that we have other crops that migrate to Africa. Maize is a big one. Manioc is another one. So we think that's more associated with this, with the 4,000 years arrival of the crop. That might be the factor explaining this decrease, rather than something linked to climate. Okay. We are not very precise. I mean, 3,000 years ago is okay. We did simulation and we show that we are able to pinpoint this kind of decrease 3,000 years ago, but not 50 or 70 years ago. I mean, the method is precise, but to a certain point in very recent time. Okay. So 3,000 years ago signature, we could find it based on simulation we did.
And another short question: as crop and human society are closely related, do you have any information about the human population size evolution during the same period?
No, but so we said, I mean, it should be possible to infer something similar based on human data. And it could be a very interesting comparison. The difficulty here for us in crop is the knowledge of the clock. So one way to compare the different crop together, we need to have a good clock. And the clock for us is mutation rate. And so we don't have a very good estimation of all the mutation rate for all the crops. So it's sometimes it's difficult to put the different layer of inference across crop on the same graph, because we are not sure we have exactly the same crop. But the pattern stays the same. The question is how do we set up where does it begin, where does it end? So one thing that is very interesting here is to then take information about archaeology. And that's what we did for Permiet. Then the inference is not based on the clock we kind of take from the literature, but that was not done on Permiet. It's based on real data and uncertainty about this data also. But certainly we could compare evolution of effective size and compare it to the evolution we have in crop. The only thing we have is generation time. So that's the difficulty. We are here in 1 year, one generation. So if we compare to human, we have a lower number of generation. And then we might hit some problem in terms of the methods, because they are effective like 2,000 generation. It's good for human inference of this kind. But if we have a low number of generation, then we are not very precise. So we might be, because of the generation time in human, we might be not be able to compare that easy between them.
Thank you. Do you have data of the prevalent cultural system with domestication of wild species in the case of rice, in wet and dry rice system? I'm particularly interested to know more about the salty, swampy rice domestication and diffusion.
So I didn't get part of the question, so could you repeat?
About the prevalent cultural system regarding the rice. Because you emphasize the importance of climate changes.
Okay.
But and you know there is a lot of hypotheses about the there is a second center of rice diffusion, that is Casamance-Gueba countries, and there is a hypothesis of what is prevalent. Is it the wet, the dry rice system, or the wet rice system in the mangrove?
Okay, so so we certainly don't have a lot of sample in our from the really mangrove, but the wild rice in northern Mali are kind of a little bit associated with pound, like water, but declining water. But I mean, most of the agriculture of rice in Africa is mainly rain-fed, but we have some area. Actually, even in Mali, where we have flooded agriculture. So we didn't include a lot, and there is other work that show that there is some peculiar diversity and in the more Guinean area. So I don't think we could say much because we didn't cover that much this range. In terms of genetic analysis. So I'm still not sure what I could answer to you. But there is so some hypothesis that there were some kind of bottleneck, also colonization of the Guinean part. And maybe that could also be linked to, I mean, what we observe, and that's very important to say that. What we observe in a lot of cases is that we have hybridization with wild population along the way of the diffusion of the plants. And sometimes new adaptation may be linked to this hybridization. So by diffusing in some area, if there is some wild relative there that have specific adaptation to growing underwater, then we could have pick up some alleles that allow these hybrids to grow in this condition. We observe some pattern that is also for permeate. We have some diversity that are a little bit very high up to India. So we know certainly that something happened there with some wild relative we don't know. And we have a kind of hotspot of diversity in the south of India. Today we don't know why, but that doesn't make it different based on the gross history from the origin, but make it important when we try to understand how the plant will adapt to future condition, because it's local adaptation that might be different from just an origin in the drier environment.
So one last question from the chat. So there is a question from Anne-Marie Chèvre. So you showed that there are many species coming from the same geographic origin. And Anne-Marie asks whether this fits with some spot of sedentarization and farming, or no idea.
What we infer is more a little bit north distribution, at least for permeate. So it's maybe area that become drier and drier and move the plant south, and with moving the population and the plant south, then some kind of agriculture develops, and the population that thrive maybe were the one that were adapting crop to agriculture and farming instead of hunter-gatherer. And so the pattern for us is more linked with the drying of the Sahara and moving south. And does agriculture become before? Does agriculture is during the process of moving? I'm not sure. But what we know is that, I mean, what we know based on our data is that permeate begin to migrate 5,000 years ago to the south. And diffusion of permeate agriculture is 5,000 years ago, based on models and archaeological data. Okay. There is lunch, so if you have other question in the room, we could discuss.
It's working now. Okay.
Thank you very much to the three of us. So Matthias Jakobsen from Uppsala University, Sweden, Hernan Burbano from UCL, University College of London, England, and Yves Vigroux from DIAD unit from CIRAD here in Montpellier. We heard IRD, sorry, here it's in Montpellier too. And so we heard very interesting speeches this morning on crop diversity to understand human and plant coevolution. So we heard a speech about demographic and genetic consequences on the development of agriculture in human population. Another one on using ancient DNA to understand crop domestication. And last, from Yves, the history of domestication to understand evolutionary dynamic of crop diversity of rice, pearl millet, yam, fonio in Africa. We covered different part of the world, from Europe, passing by North America, and finally Africa. And we covered also a large scale of time, from Neolithic era to domestication, agriculture domestication 2,000 years ago, and more recently. So it was a large spectrum of data on crop diversity. I hope you enjoyed it. Sorry. And we'll now just go for lunch. I just remind you that there is a poster exhibition in the hall downstairs, and we'll go back to the second session of the day at 1:40 to learn about how can we use crop diversity data to face actual global challenges. So we will move from ancient time to nowadays and talk about global challenges. So thank you, everyone. Thank you to the speakers from this morning, and see you at 1:40. Thank you. We won't close the amphitheater, so just make
Hello everyone, let's go back to our business. So now we'll see this afternoon for this second session of this Arcad meetings scientific seminar, this session dedicated to ongoing transitions. So we undertitled it Crop Diversity as a Leverage to Adapt to Global Changes. So we'll hear some high quality panelists, Victoria Reyes Garcia, Njido Kane, and Bruno Fadi. And I'm just giving the floor to Christian Leclerc, which will be the chairperson for this session. Thank you very much.
Ladies and gentlemen, good afternoon. Welcome to the second session. My name is Christian Leclerc. I'm a social anthropologist. It's a real pleasure to be the chairman of this session with Adeline.
Hi, I'm Adeline Barneau. I'm a population geneticist, and I'm very pleased to be with you today.
The aim of the seminar is to draw a panorama of crop diversity studies. And after the session this morning, we take into account the past transition. We will examine this afternoon how diversity can be help response to the current transition due to climate change. Everyone here, at least me and Adeline, absolutely, we believe that the crop diversity dynamic cannot be explained or described only referring to biological factor, but absolutely also to social and historical factor. Through the three case studies this afternoon, the three keynote speakers will present the social and the genetic dimension of the ongoing transition to phase two global change. I will ask to Victoria Reyes Garcia to come here with us. Victoria is an ecological anthropologist. We can say that, huh? From the Institute of Environmental Science and Technology in Barcelona. And Victoria is coordinating a project, a ERC project, LICHI, that deal with the local indicator of climate change impact and take particularly attention to the local knowledge to phase two climate change focusing on local community. Victoria will take us through what adaptation to climate change means for small-scale agriculture. Thank you, Victoria.
So thank you, Christian, for the presentation, and thank you everybody, the organizers of the Arcad conference, for inviting me to give this speech. It has been maybe 2 years since I spoke to the public, like today with the COVID. My last speeches were always on the screen, so I hope I can still hold your attention. So as Christian said, I'm going to be talking from a more social science perspective and trying I have prepared that talk having in mind a more like scientific or agronomic perspective. I hope I hope it's still interesting for you. So let me start. Can I pass from here? So let me just start with some very basic premises that are going to frame the work. So the first idea that we all need to keep in mind is that there is no country, no place on earth that is not exposed is not experience climate change. And I'm not going to try to explain climate change or to try to convince you that climate change is something that it's happening. But what is important from the perspective that I want to talk today is that the effects of climate change are not the same for everybody. They depend a lot on where you are living. So it's not the same if you live in the Arctic, right, and you feel the glaciers melting, that if you live in the islands of the Pacific and then you experience sea level rise. And this is important because we always hear this idea of like 1.5 degrees Celsius increase, but 1.5 is an average, and for some people are experiencing 3 degrees increase, and some people are still not experiencing that, and some people are facing rains, and some more rains or extreme events, and some people drought. So it's important to keep in mind this diversity. The other thing that we are still in the premises, in the kind of like contextual of this presentation, the other thing that it's important from the perspective I'm talking is that beyond the atmospheric and the biophysical impacts, climate change is having all these cascading effects that go to the biological systems, right? So the impacts of climate change are not temperature. And in our work, when we talk to indigenous people and local communities, most of the impacts that people perceive relate to changes in the biological systems and in the human systems, right? Changes in the time of the flowers of the seasons, changes in the time that the birds come, the fact that some mismatches between the flowering and the pollinators. These are the types of things that people perceive, and they don't perceive as much that it is a little bit hotter. They do also, but these other biological impacts are the ones that are really taking the stage. And it's also important to kind of like keep in mind that some of these impacts that we talk are sudden. They just happen from one day to the other, like the extreme events, and some are incremental, right? So there is a incremental change in temperature that little by little change the behavior of people. And finally, the last premise that I wanted to, it's like all this introduction before I get to the point. But the last important point is that for rural and small farmers, climate change, it just adds to many other drivers of change and to many other stressors that they have in their life. Here, this is a matrix of some work that we did with the Chimane, which are an agriculturalist, hunter-gatherer, becoming agriculturalist group in Bolivia. And we were asking, what are the drivers of the changes you see? And in blue here, you see drivers that has to do with the climatic system. Well, you can actually not say it, but if you believe me, that what it says there is changes in rain generate that there is less water in the river, that affect the fishes, that make that we cannot have enough fishing, right? But they also say there is population growth, there are changes in the policies, there are overfishing, and all those things also affect our crop system, our fishing systems. So people don't perceive climate change as the only driver. They see it, they perceive it, but they see it in interaction with these many other drivers that are there affecting their systems. And just recently, the IPCC and the IPBES, they have put together this workshop, this coordinated workshop with IPCC and IPBES, trying to see, okay, we are these two bodies of policymakers and scientists working separately, and actually we need to be working together, because what happened in the climatic system affect the biological and the human systems, and vice versa, right? And this is something that that indigenous people or local communities also tell us. So because there are all these changes and all these impacts, people adapt to those. Adapt, it means that they respond to these changes. There are changes happening in their system, and they adjust by doing adjustment in their way of living, in their socio-ecological systems that help them survive in the new situation. We differentiate or the literature differentiate between planned adaptation, which is when the state does something for you, and autonomous adaptation, which is what I'm going to be talking about, which is people change something in their behavior based in what they know, in the capacities that they have to actually adapt to their new situation. And crop diversity is actually one of the main focus that have been in these autonomous adaptations, right? Farmers adapt to to more drought by switching to crops that might adapt, no? So these are autonomous and spontaneous adaptations that are not given, but that people develop through time. I also want to emphasize that this idea of adaptation varies largely, and it depends completely on what are the resources that people have, what are the existing institutions, what are your capacity. So some people, if we take again the example of sea level rise, people in the Fiji Islands adapt by actually complaining and protesting and saying, guys, we are getting drowned. And people in Australia, I think these pictures are from, I don't remember, but they just adapt by building big infrastructure, right? So adaptation can have two very different forms depending on what are your resources. And also it's important to think, or from the social science perspective, when we think of adaptation, we need to think of that as a process, right? It's not that something is adaptive and it's okay, we manage, we have now like the crop resistant varieties, we have adapted, we are all fine. No. You start with a process of adaptation that you mean to actually be better, and it can become just a coping strategy that leaves you in the same place, no? Because then the rains continue without coming, and then the crops are not good anymore. You can also, and some of these adaptations, or some of these responses, let's call it, can become maladaptive, right? You just did something, you just adopted some strategy to actually to face a risk, a climatic risk, and in the first time it worked, right? But then you just like go into a maladaptive outcome because this was not sustainable in the long term. So this is very important to keep in mind, because adaptation is not something that happens now and it's solved, right? It can work for a period of time, but then over time, don't actually work. One example, this is just from someone else's work, is this example in Ghana, where like farmers, they migrate out of the area because with the rains there is no actual labor, or they cannot stay in their place to work. At the moment, in that moment, that helped to adapt because it diversifies the income, and you think, oh, that's good. They have more sources of income, they have adapted. But then the following year, when the rains are again okay, there is no labor force in the village to actually maintain the crops. So actually, it becomes a maladaptive situation because the household doesn't have these different sources of income. I'm not going to get into this graph, but I just wanted to show that we have done this with Anne Slingman. She's a student working in the project that Christian was presenting, the LICHI project. We have done this huge literature review on how farmers and local communities and indigenous people in general, what are the autonomous adaptation strategies that they have found. And we have done all this like work of classifying. It depends on their livelihood, it depends on where they live. Some of them adapt by changing their way of life, some of them adapt by migrating, etc. No? So I mean, we have done like, this is a quantitative review with all the different forms of autonomous adaptation. The first important thing regarding farmers is that they don't adapt through crop diversity, they adapt through mostly, like when you look at the numbers, right, they mostly adapt with other household assets. They rely on social capital, they rely in changes in infrastructure, they use other strategies that do not relate to the farming system to actually face the climate change impacts. However, they do also use many strategies. All this comes from a literature review, right? They also use many strategies and local responses in the cultivation sector. They actually, the most common one, it is indeed changes in crop composition. So farmers do change the crops in their fields to adapt to climatic changes. They also changes cultivation practices, like for example, the soil conservation methods or other practices. And they also do changes in the application of productive resources. No, they use more irrigation, they use more fertilizers. However, although, oh no, sorry, I have changed my my presentation in last minute, so not yet. However, so because of the results of this research, there has been all these, I mean, there is all this interest in researchers in the field looking at how agrobiodiversity and climate change relate, no? And most of the research have been saying, as Christian was saying at the beginning, agrobiodiversity is good to adapt to climate change because it helps with farm resilience, crop diversity is a good base for adaptation, it helps with food and nutrition, right? In front of these changes with the climate change. And here we are going to be looking at that through the lenses of the smallholders, farmers, right? The small farmers. Because most of this research has been done from a more biological perspective. And I want to take you through a trip through the lenses of the farmers. Let me just say that another literature review that has been conducted in the area looking at what are the crops that are adopted for climate change, it shows that most of the adoption are in the sub-Saharan Africa are for crops that are more drought tolerant and that are more water use efficient, right? So there is not a lot of adaptation, not a lot of people who adapt through changes in crops, as we said before, but those who do the crops that are preferred are these drought tolerant crops. And these adoptions depend on many factors according to this literature review. And some of the factors that are the most significant in this adaptation are whether the extension services are effective, what are the education level, what is the access to inputs. So if you look at this graph, you can see that farmers that are adopting those crops are actually farmers who have some kind of like income level or some access to these services. But many of the people in the world don't have, or they depend on those. So in the last slides, I wanna talk a little bit more on how does all this look in the field, right? Because all these are literature reviews and people, I mean, more high level. But in the field, when you go and ask to the farmers, what are you doing with your crops to adapt to climate change? How does it work here? It looks completely different, right? And this is what I'm gonna just present now, is on a case study that Ana Porcuna, who's sitting there, very shy. This is actually her work in Senegal, in the Basari country. I only have 5 minutes, so I'm just gonna maybe go fast on this in this part. The Basari is tropical, dry climate, subsistence farmers, and they are experiencing a lot the climate change, basically on the shortening of and the delay of the rainy season, and also a temperature increase, like in many places of tropical area. The Basari, I'm not going to get into the graph, but the Basari grow all this diversity of both at the landscape level, right? They have different, like in the hilly areas, they have more traditional crops like fonio, peanut, sorghum. In the more low areas, they have rice and maize. So they use the diversity of their landscape to grow different crops that serve them for different purposes. So they use this diversity. They also use a lot this diversity at the crop level. They use this is an example of sorghum. So they have several sorghum varieties that they grow, and we ask them. Well, Anna asked them these different sorghum varieties, what are good for. And if you see the spider on the side, every color is a different variety of sorghum, and the peaks there are different traits, no? So some of the varieties they want because they have good taste, but the ones that have good taste, maybe they cannot sell or they don't sell. And the ones that have early maturity, they are bad for soils or for some other thing. So when you look at the combination of the different varieties and traits, you see that they are actually using all this diversity to fill the preferences, but not only agronomic, also in terms of taste, culture. They use some of them in rituals, others not. So there is all these varieties. So it's like, okay, it's good. I mean, these people are actually using diversity to fulfill all these purposes. But when we look, this is what, if you look at only at today, but when you look at the more temporal dimension, as I was trying to say, that adaptation has to be looked through time, what we realize is that in the 50s, the Vasari have all these drought-tolerant varieties only. Sorghum, I mean, not varieties, crops. Sorghum, Bambara nut, phonium. In the 70s, they increased the diversity, and this is what we see now. They increased the diversity because agricultural services came and gave them peanuts and maize and rice, and now they can get water with water pumps and have more diverse land, more diversified agricultural system. But now, and this is also part of the work that Anna is doing, they are abandoning the drought-tolerant crops because drought-tolerant crops have much more workload, because the kids don't like that, they like maize and rice, because the extension program are giving them free maize and rice varieties. So it's actually switching to this other side of the graph. So the question here, or like the point that we are looking is like, okay, if you look at a snapshot of the agricultural system of these people, you can think, oh, this is a very diversified system, no? The ball is on the top, that's what we're seeing now. But when you look through time and you look with this perspective of adaptation and what crops they are being selected and what crops are being abandoned, then you might find yourself in these different pathways that I was showing you. They might be maladapting because they were selecting crops that now work well and give them income because it's a short term, but in the future are not going to be adaptive to a drought condition. No? So this is like the point of looking at this temporally. So let me just to conclude. I mean, the whole point I wanted to do with this long and I hope not so boring presentation is that small scale farmers, they face simultaneous and interlinked stressors. They don't adapt to climate change. They adapt to new situations generated by climate change, political change, tourists arriving, new road, losses of biodiversity, land grabbing. They adapt to all these that affect them in a synergistic way. And they provide many responses to those stressors. And some of these responses relate to their agricultural systems and some others don't. And we cannot forget that farmers that migrate, it's just also like a response, but it stops being a farmer, but it has to be considered also, no? And also that not all the responses that people do when facing climate change, not even if it's increasing agrobiodiversity in their fields, not all of them are adaptive. You have to look at that through the lenses of time, because over time can be maladaptive. So if you have been sleeping or bored or whatever, just please now take attention. This is the take-home message. If you just hear to this last one slide, I'm fine. So the take-home message is that to understand these small-scale farmer responses to climate change impacts requires this contextual analysis that includes this long-term temporal view and the consideration of other stressors that go beyond climate change. We cannot focus only on climate change because people respond to many things and respond over time, and we need like a longer view. And with this, I hope I didn't pass my time, but thank you.
Thank Thank you, Victoria. Diversity of situation, diversity of local indicators, diversity of way to face to climate change, I mean, not only. Do somebody have a question to Victoria in the room? Yes.
Thank you for the very interesting distinction between short-term and long-term adaptation, and to stress the opportunistic strategies of a few actors and farmers. I believe you have done the interviews at the household level. I would be interested to know more about the methodology. And on another point, there is a new project. I'm not sure Frédéric Jankowski is in the room, but and the title of the new program of research is Seed Attach. And I am wondering, on the long-term strategy, don't you think that there is also to take into account not only the economic opportunities, but also all the cultural drivers and the importance of attachment to specific seeds in terms of heritage for the food and for, I made the comparison with other countries and where rice is supposed to have been declined or abandoned, such in the Salomon Islands. It is not the case because the mothers, they need rice, although the climate conditions are absolutely not adapted, and they conserve the species of the seed because they need it for specific use and ritual use. Thank you.
Yeah, thank you for the question. Regarding the first one on the methods, yes, we did we do these surveys at the individual level, not even at the household, so it's the individual. And the questions, I mean, for the particular case of the Basari, Anna is the boss, so maybe later you can talk to her. But basically, we do questions on what are the changes in the environment that you perceive, what are the drivers that you think, what's driving this change, and then we ask them, which seeds do you have in the field? How long have you had this seed? Where did you got it from? Why that seed that you had in the past? Which seeds did you had? Why did you stop growing it? And so it's kind of like methods of like trying to map over time all the seed diversity in the different fields. Regarding the issue of the seed attachment, yeah, as I say when I was showing the spider, there are many values that people hold. I mean, that the seeds hold many values and many knowledge, and I mean, every seed is kept for different purposes. But here again, it's important to look at this in a diachronic perspective, because I mean, and also again, in this particular case, and I can speak more from that than me, but even if the older generations are saying, yes, I'm keeping these, and this is what it's important for the ritual, these sorghum varieties for the rituals, and we need to keep them, even if it's only a little bit. Over time, like if you look at this in a 20 years perspective, their kids don't want to eat sorghum anymore, they want to eat the rice. So eventually you could see how when the old generation, and it might not be true for all varieties, right? Some specific varieties might stay on the system. There is some stronger attachment in one than another. But if you look at the diversity of crops, small crops in the world, the answer is there, right? We have reduced the diversity of crops that people grow in general, and there are all these substitutions. So obviously, some are strong, some stay strong, and this is something that can help, actually. That's something that we could use to help maintain varieties, the fact that there is this attachment, and it's real, and I think, but when you look at the system and in a big scale, actually, you see that the tendency is not that one, even if there are exceptions. Yeah. I think there was another question. No?
Yeah. Me, I have a question that is related to the seed attachment, but not related to seeds, related to knowledge. As we usually said, there is traditional knowledge, And the adaptation of these populations are usually based on the traditional knowledge that is transmitted from generation to generation. And we are, sometimes we believe that the tradition or traditional knowledge is something that is not evolved too much from one generation to other. How do you conciliate traditional knowledge and adaptation?
Right. So the traditional knowledge, in my view, or in the view of those of us who work on that, it's a dynamic system, right? And the whole point is not the knowledge in itself, but what do you use this knowledge for, and what's your relationship with the environment, right? So, for example, in the case of the Basari, they used to be hunter-gatherers until the 50s or something, when they did the national park, the that park, and they were expelled, so they start farming. So they have lost that particular knowledge. They might have lost a lot of the knowledge that they use for the hunting, gathering, and this particular environment, but because they still continue to depend on nature, they have developed a new traditional or local knowledge more related to the agriculture and including some of the knowledge that they have of wild edibles, etc. So it's a dynamic system that allows them to stay in the environment. If the environment changes, they change their knowledge and they use their basis to adapt. But the problem is when you don't let them keep their, well, don't let, when the varieties that they use are not grown by themselves, but they are brought by the agricultural services, then the knowledge that it's embedded in this variety, it's also not their own knowledge. It comes from somewhere else. So it's very dynamic.
Thank you, Victoria. One last question.
I had a quick one. You mentioned the difference between sort of institutional adaptation and spontaneous adaptation from the farmers. And would you say that if you consider institutional adaptation, there is cases of maladaptation as well?
Oh, yes. Yeah. Because, I mean, I don't have an example now from the agricultural system, but from for example, when they construct barriers to avoid to avoid the effects of sea level rise in Fiji, right? They do all these barriers in the coast to avoid that water enter into the agricultural fields, because with sea level rise, then the fields get salt and they cannot produce. So they build these barriers. But of course, these have consequences down, or consequences for the fishing, or so some action that the government did to avoid that water was entering into the agricultural field actually also had bad impacts for the fishing and for other sectors. So yeah, I mean, obviously plan adaptation, plan responses cannot be adapted, or are not always adapted.
Thank you very much, Victoria.
Thank you all.
And now we are happy to welcome our second speaker, Jido Khan. Jido Khan is a plant geneticist, a molecular biologist, and is a director of a regional study center for improvement of adaptation to drought, CERAS, in Senegal. And this center was recently awarded Regional Center of Excellence. And now Jido is proposing another case study concerning Senegal biodiversity and agrobiodiversity adaptation towards strategy and innovation developed under drought-prone environments. So are you here, Jido? Can you?
Yes, I'm here, and good afternoon to everybody. I hope you can hear me. Good afternoon. I share my bulletin from the Senegalese Agri-Research Institute. It will be easier. And I will share with you first the strategy.
So welcome, Jido. The floor is yours.
Good afternoon. My name is Jido Ken from the Senegalese Agri-Research Institute. And I will share with you first the strategy Senegal is engaged, and in second, the crop innovation we are developing a response to drought-prone environment. Around 8,000 species have been inventoried, including 174 cultivated varieties of crop composed of cereals, vegetables, and fruits to feed a galloping population.
Guido, can you hear us?
If you can hear me, I can make it live.
Yes.
Okay. So I will share my screen.
Yes.
Good afternoon. My name is Guido Ken from the Senegalese Agri Research Institute, and I will share with you first the strategy Senegal is engaged.
Guido,
Second, the crop innovation we are developing a response to drought-prone environment. I can make it live. But it's okay. Can you hear me?
Yes, we can.
Okay, I will just share the strategy that we have put here in Senegal and some crop innovations that we have developed at Serras under drought-prone environment. Good afternoon. My name is. Sorry, I have to change the presentation because this one I recorded. Okay. Yes. So rapidly, I will just present you the diversity here in almost 8,000 species that we have inventory, which consists of 174 variety of cereal, edible legumes, and vegetables. And we distinguish also 6 agroagricultural area, which is composed of 700 costs of cost, and the growing population of basically our national strategy and action plans envision to restore, conserve, and exploit biodiversity for sustainable use, socioeconomics, and 13. So this strategy will implement it and have 4 axes. The first one is to preserve the reduction of biodiversity and power institutional capacities. We also have to engage at the public to take into account biodiversity into their strategy. And last also, it was, yes.
Guido, excuse-moi, est-ce que tu peux couper ta caméra pour qu'on t'entende mieux? Merci.
C'est bon?
Oui. Merci. Guido, tu nous entends?
Je pense que la connexion n'est pas bonne. Si vous pouvez jouer peut-être l'enregistrement envoyé.
Oui, on peut faire ça.
Désolé.
Mais non, c'est nous qui sommes désolés.
engage. And in second, the crop innovation, we are developing a response to drought-prone environment. Good afternoon. My name is Jido Ken from the Senegalese Agri-Research Institute, and I will share with you first the strategy Senegal is engaged, and in second, the crop innovation, we are developing a response to drought-prone environment. Around 8,000 species have been inventoried, including 174 cultivated varieties of crop composed of cereals, vegetables, and fruits to feed a galloping population. Adeline, je propose que si le prochain intervenant enregistré dans 5 minutes.
Oui, ça me...
Pour voir le problème de connexion. What is the status of plant genetic resource management? Pourquoi ça ne marche pas? There is two conservation methods. In situ, which consists on farmers' field, protected area, classified forest.
Je vais les introduire.
Thank you, Jido. We are sorry for the technical problem we face too. I don't know if Bruno Fadi is online.
Yes, I'm online.
Welcome to you. You see, we have some technical problem with the connection with the Mali, I guess, where Njino is. We will ask you if it's possible to do your presentation now.
Sure.
Okay, can you share your screen on Zoom? Thank you very much. I will only introduce you while you are preparing the screen. Bruno is working in forest ecology and genetic. He's a researcher in the research unit in ecology in the Mediterranean forest in Avignon. He's the coordinator of European H 2020 Gen 3 project, which aim at optimizing the management and the sustainable use of forest genetic resources in Europe. I would like to welcome you, Bruno, and the floor is yours. Could you changer le mode de présentation? Tu es en mode de présentateur.
Okay.
Thank you. C'est en haut, voilà, paramètres d'affichage.
Sauf que, attends, je vois pas.
Juste le deuxième depuis la gauche, paramètres d'affichage. Là où tu es, en haut. Ouais.
Sauf que moi, je vois pas afficher la barre. Paramètres d'affichage.
Voilà. C'est pas ça. Voilà. Thank
Là,
you.
vous voyez comme il faut?
Tout à fait. Thank you.
Okay, great. All right, so good afternoon, ladies and gentlemen. Thank you for the invitation and the presentation of who I am. I'm going to talk to you about about forest trees this afternoon. I will first set the scene, then talk about genetic consequences of what it means to be a tree, and then focus on a gene conservation program that is going on in France and coordinated throughout Europe. So the first setting the scene, the forest. It's about 31% of the global land area, so it's a bit less than agriculture, but not too much less. They are unevenly distributed on the globe, as you can see from this map. And one of the things we can say about forests right now is that they are at risk. FAO has listed throughout the years a decrease in land area, and mostly this decrease is due to land conservation to agriculture. So somehow agriculture and forest conflict, although they should not. In Europe, it's about the same ratio between forest and agriculture, 42% forest in the EU and about 48% agricultural land in the EU. So it's about balanced. Europe is actually quite a forested area. However, if you take a long-term perspective, as we were asked to do by the first speaker, the potentiality for Europe in terms of forest, if you look at what happened at the onset of the Holocene about 15 to 20,000 years ago, it is 80%. So actually, man, throughout his activities, has reduced forest from 80% to 42% of European land cover. A few things more about forests. I will mostly be talking about natural forests, naturally regenerating forests, and there has been ongoing discussions about whether or not plantation forests are forests. But as you can see from this graph, the yellow part of the bars, they actually are few in number compared to the overall amount of forest. It's only 7% of all forest across the globe. And in the EU, it's also very few. So what is a forest? According to FAO, which also has been discussed by many NGOs, it's a land spanning more than 0.5 hectares with trees higher than 5 meters and a canopy cover of more than 10%. What is a tree, actually? There also has been discussion about what is a tree. So this is a paper about what is a tree in the context of the Mediterranean basin in Europe. It has consequences. So we should define what a tree is. So it's a woody plant that's tall. So FAO says 5 meters. In this review, we say 3 meters. So whatever. It's a tall woody plant which has a trunk. It lives in a forest, and the next points are important for gene diversity. They are long-lived. They have an extra long juvenile period during which they don't reproduce. When they reproduce, they usually have a massive seed production, either annually or as a masting system once every 5-10 years. And population sizes are usually very large. So the consequences for gene diversity and evolution is that those forestry populations have a huge gene flow. So they exchange genes through gene flow, mostly through wind and animal pollen propagation, but also through seeds. Their mutation rate is slow. Their speciation rate is slow. I mean, if you think about it, there are species in North America that still hybridize with European species after 65 million years of being separated. So speciation rate is low. There are large effective population sizes, meaning a large potential for evolution. There are demonstrations of rapid local adaptation and differentiation for many traits, for example, adaptation to light periods from the equator to the poles. And there also is often demonstration of high phenotypic plasticity. So it means that forestry populations have high genetic diversity within populations, but also significant differentiation for many traits. Of course, exceptions exist, and this is a picture of Italian stone pine, Pinus pinea, from the Mediterranean basin, where although there is huge phenotypic plasticity, there is absolutely no genetic diversity, and this can be explained, and we can talk about that in the discussion. So because of these traits, and because of those traits and population traits influence genetic diversity, the framework for protecting those trees has been placed mostly on in situ gene conservation, and ex situ strategies have only been used when the species or the populations are endangered. What I'm going to say, I must be specific about it. This potential for in situ adaptation works only under the Paris Agreement on climate change. If global temperatures rise over 2 degrees, I think what I'm going to say here doesn't work. We've had numerous EU-funded projects to deal with this in situ gene conservation. The first one of GIS is an information system. Gentry is a project I coordinated, which ended in 2020. The GenRes Bridge project is pulling together people from animal, crop, and forest genetic resource conservation to build a common framework. And 4Genius is a project that just started, which analyzes the diversity of gene conservation units in Europe. The political framework for this is, first of all, the overarching Convention on Biological Diversity, 1992. And at about the same time in the 90s, the European governments got together and formed the Forest Europe Ministerial Conference for the protection of forests in Europe, meaning sustainable management. From this emerged, to the bottom right, the EUFORGEN program, which is a specific program for gene conservation and genetic resources management across Europe. And in France, we have a commission on forest genetic resources with a legal and administrative framework to set up conservation units. So I will go now with an example of a tree species for which there is both in situ and ex situ conservation because of different reasons. It's a species well known to people from Montpellier. It grows in the Cévennes region and in the Pyrenees on the Mediterranean side of the Pyrenees. It's a local subspecies of black pine, Pinus nigra salzmani. It's protected in Europe under the Natura 2000 framework. And its main threats are fires, land use change because of clearing for agriculture, logging, and hybridization with massively planted other black pines as forestry species. So to build these networks of in situ conservation and ex situ collections, what do we need? What do we usually use in forestry? Well, it's good to have a phylogeny. So, for example, in pines, the phylogeny is quite well resolved, and we can place our species within this phylogeny and start building our strategy from there. Then it's good to have a distribution map, and EUFORGEN has been essential in providing distribution maps of most European tree species, forest tree species. So here we have a tree species, black pine, European black pine, and 5 subspecies, rather well delineated. Now, the question that we have to ask is, do these subspecies, including the salzmani one in red, do they differ from each other from an evolutionary point of view? Do they have different adaptive traits? Can we characterize them? And then can we build a network from these characterizations? So in black pine, in France and in Europe, we have a network of common gardens where different geographic origins are compared for different traits. So here, for example, we measured the trunk diameter and tree ring width. And we look at how different different origins, different provenances. So it's on the bottom part of the figure, it's the horizontal lines, how they differ with their reaction to environmental drivers. And here it's maximum temperature. And we see plasticities, the 4 blocks correspond to different common gardens in different places in France. And you see positive and negative reactions, positive green, negative yellow, orange. So this is one way to characterize. Growth is usually a good trait for fitness. Then using molecular markers, we can look at at evolution, at differentiation. We can see if the classical botanical differences measured by anatomical and morphological traits actually correspond to something that makes sense from the genetics point of view. And here we have discrepancies, but we see that basically we have lineages that fall not too badly with the classical view of taxonomy, except that the last green part, which is in Cyprus, should be probably something that is pushed to the rank of subspecies. And then we fit coalescent demographic scenarios, and we explain when this differentiation took place. And in the case of black pine, we were surprised to see that it is a very recent, actually, differentiation, sometime at the end of the last glacial maximum and during the Holocene. You can also see in the species that there is extensive admixture in the little groups with the colored bars. You also find mixtures of different colors indicating admixture. Okay, so using this, you can start building your conservation units. And in black pine, the difficulty is that it is difficult to find which populations, which forests locally are actually autochthonous because of substantial forestry activities and plantations. So in France, plantations started sometime in the 1860s, and they were extensive, particularly using black pine. So using both historical documents such as this one and wood cores, which can tell us the age of trees, we cored potentially autochthonous populations, and we kept for analysis only those trees that were born before the heavy plantation started in 1860. So that's a good thing with trees. You can use wood cores to actually date your sampling. So here are a few of those populations where the red dots are, the historical photos of 1900 and what they look like now. So we could pretty much see that actually they existed at that time. So this is in the lower Cevennes region. This is in the Pyrenees. This is the well-known population of Saint-Guilhem-le-Désert just north of Montpellier. And an interesting population to sample on cliffs. Interesting for technical teams, actually. So with all of this, we collected our samples and looked at the regional diversity and structure of our population in France. And we found actually 6 lineages, also post-Holocene ones, with admixture, as you can see from the colored bar chart. But we could pretty well separate those into regionally meaningful groups, with the 4 groups in the 5 groups are in the Cevennes and 1 in the Pyrenees. So by this, you can actually start designing your conservation units. So designing conservation units, we use a reference document that was published by people from Ufergen in 2011. And this describes how you do a conservation unit, what you should have, the minimum requirements, what the size of it should be, and so on and so forth. So I can explain that more and the constraints with the local management that they entails. But to show you an example, here is the most recent of our conservation units for black pine in the Ardèche region, north of Montpellier. And we have a core center where protection is maximum, and then a peripheral region around the center of the conservation unit, which acts as a buffer against gene flow. Right. So for black pine, as I told you, we set a two-way approach: the in situ one, which I just described, and then the ex situ one, because of extensive risks faced by this subspecies in France. So for this, what we do, I think it's the same also for agriculture. We collect grafts or cuttings. Here it's grafts, and we plant them in a collection. So this is our core collection in near Marseille in southern France. And this core collection with grafted clones can also be used as a tree seed producing orchard for diversification, pending the following of recommendations and guidelines. Right, so with this, I would like to end my talk by showing you how we're doing in terms of gene conservation units in Europe and in France. So at bottom right, we have, these are all the conservation units that have been set in situ following the way I described. We have that for over 10 species, 10 major species for forestry in France. And in Europe, we have the map top left. These are all the dots where represent in situ conservation units coordinated by U4Gen, accessible through this portal. And these indicate the efforts that countries do in terms of gene conservation units and in situ conservation in their country. So many, many dots and something around 100 tree species, which I think is pretty good. Finally, I would say that the way forward now is, as high-ranking officials said at the closing of the World Nature Congress in Marseille, beginning of September, it's not enough to have conservation done on paper. It's important to have conservation done in the field and actually having people who manage and monitor. So our way forward with the U4Gen and the French In situ conservation network is to decide on how we monitor. And there has been extensive discussion about how do you do monitoring. The fact that it is important to include it in the CBD post-2020 framework, which is coming up, because you probably know that, but genetic diversity is poorly taken into consideration in the CBD, which mostly focuses on biodiversity at species and ecosystem level, but often forgets that genetic diversity is the basis for evolution and adaptation. So for us in forestry species, this is where we are now. We are gaining momentum. We have quite a few links with nature conservation, and we actually need to keep the identity and specificity of gene conservation recognized as such and beyond immediate usefulness. With this, I will close my screen, and I thank you very much for your attention.
Thank you very much, Bruno. It's very interesting to see the contrast between crop, annual crop, for example, and tree. And for us, I use a sentence you said, Bruno, evolutionary consequence of being a tree. It could be evolutionary consequence of being a human. but it could be also evolutionary consequence of being a tree when human is using a tree. It's my first question, just to start the question to you. What do you have a bit more precision on what is the consequence of human exploitation of the forest over time? And the second question is the local implication or participation of people that are living close to the forest. Is your management plan or conservation plan or construct with people in order to work together with people that are living close to forests? Thank you.
Right. So the global trend with forests is a decreasing one. The FAO has done this world forest assessment over the years. I don't remember if it's the 4th or the 5th assessment, but the forests are decreasing globally, but there are huge differences. For example, in Europe, they're increasing in size and in cover. Of course, they're not old growth forests, but they're new forests, but still the trend is increasing. But as FAO mentioned, the decreasing trend is decreasing. So it's like, it's not accelerating at any rate. So that's the scene. Now for management, I totally agree that the human factor is important. We never design conservation units in a place where we have opposition from management. So in France, mostly the gene conservation units are in public forests. And we have the support from the local management, and often we are in community forest, community-owned forest, where the municipal council actually signs an agreement and says, yes, we want our forest to be used as a gene conservation unit. So yeah, the human factor is absolutely indispensable. And as I am responsible for silver fir and Norway spruce networks, and I would never recommend the setting up of a conservation unit in a place where people don't want to do it. It's counterproductive.
Thank you. Is there another question, Jacques? Yeah.
Hi Bruno, nice to see you.
Hi.
Jacques Davis speaking. I was wondering, you said that your plan could work until, I'd say, 2 degrees of warming. What will happen if it's more? And do you think that the in situ population that you have think or planned will still be working, or do you think that by any way you will go to the debate of assisted migration? And so what are your, if we see it dynamically, do you have debates in the forest community about ways to make the genes to migrate more northern or more in altitude? So with all the debates that we could have about the perturbation of any kind when you migrate species to you. So we often have such discussions for the wild relatives of crops, and we think that definitely you should have think about this much more than we have. So please.
Right. So I don't know how much time I have, but this could lead us to a very, very long discussion, and probably not everyone will agree. But so if I can say something briefly. Yeah, 2 degrees. I think over 2 degrees, as pointed out by the IPCC, major changes will happen, and well, adaptation has limits. So I expect that many parts of our network would not be under suitable conditions anymore, and species would be replaced by others. Okay, so to give you an example of what's going on right now, so we have approximately a 1 degree warming since when the trees were born, actually, in the 1800s. And I was called by people in the Jura Mountains, so an environment that's not very warm, to discuss about the fact that one of our conservation units was actually declining, and should we not change it somewhere else? Right, so we get there, and indeed the adult trees are dying, but we see quite a number of seedlings in there. So after much discussion, it was agreed by the manager to not do what they would usually do in a case like this, which would be to change completely their objective and use another species, but actually help those seedlings that existed to grow and to have a management in favor of those seedlings, expecting that those seedlings born under adverse conditions could actually carry some novelty in terms of adaptation to a different climate. So that's one mechanism that we're definitely hoping will happen. And in all of our conservation units where we have decline of adult trees and somehow everyday management would replace them by something else, we say, no, wait a minute, let's manage in favor of a new crop of those trees which might have a different genetic makeup and then adapt to the new climate. But if the new climate is something that has +3, +4, +5, or +6 degrees compared to now, I think they won't make it. So second strategy is ex situ. The forestry community has to do that more. We've had, since the 1990s, we've essentially pushed for in situ conservation, and we need to do more of ex situ conservation, meaning collecting seeds, putting them in seed vaults, or putting them in ex situ orchards like I showed you, where they can be a collection, but they can also be used as seed producing orchards. Assisted migration, yes, foresters want to do it. They mostly are thinking about moving species, but not using the within species mechanism of creating diversity by mixing, which is something we would recommend as a better solution right now, instead of changing completely the species and including exotic crop species like Douglas fir. But it's an ongoing discussion where the tendency would be to bring in new exotic species instead of using the natural variation that exists within autochthonous species, because of the fear that foresters have of climate change. And this fear is fueled by those nice maps that we are producing using niche models, which actually don't include any consideration of adaptation or genetic diversity. So yeah, but France doesn't plant too much. So mostly it's natural regeneration. As I showed, it's globally it's 7%. In France, it's even less. So there is, the road is open for within species use of diversity through gene flow. But we are encouraging experiments, and we're encouraging writing down what you're doing and sharing your experience with others, because there's nothing worse than doing something and not knowing why it works or why it fails. So that's also, for me, an important aspect of things. I can digress more, but I think time is running, and maybe we can have this discussion again tomorrow during the roundtable.
Thank you, Bruno. The time is going on. I would like to thank you for your presentation and the discussion. We have time, it's tomorrow during the round table, effectively to discuss and even to ask a new question to you. Thank you very much. I will propose to the room to take the break now. We have a technical problem with the presentation of Njido. So we take the break for 20 minutes up to 3:15. And we will manage this problem technical, and we will back with Gino, we hope, by 3:15, just to finalize this session before starting the last session of the day. Thank you very much.
We're waiting for face-to-face people to come back to the room. All right. Do you see me on YouTube on streaming? Is this okay? All right. So, dear colleagues, we just we'll just start our third session of the afternoon towards future transition. We will hear first Njido Kane from the director of the ISRA Serras in Senegal, who will have a speech on strategy and innovation developed under drought-prone environments. Unfortunately, we had some connection problems earlier, but now Njido is with us, so he will be live speaking. Then we will hear Ruben Mila, which is with us in the room here from University Rey Juan Carlos in Spain. Who will talk on ecological look at crop evolution. Then Salvatore Ceccarelli, sorry for the pronunciation, from Bioversity International, based in Rome, Italy, will talk on agroecology, contribution on evolutionary breeding. Then Carl Zimmerer from Pennsylvania State University on crop diversity and food, and at the end, Thaura Gnaim Herrera from the University of Cali, Colombia, about genome editing as a new source of diversity and the risk associated. So earlier in the afternoon, we heard Victoria and Bruno talking about first adaptation in small-scale agricultural system. We learned that adaptation is a process, and we have to look at it as a process. And a small-scale agricultural system, high diversity, is a local response used to adapt. We heard also Bruno Fadi from INRAE talking us about the genetic diversity of forest, and mostly on black pine, with this example in France and in Europe. And he talked on in situ approach and ex situ approach, and the complementarity of those two approach, and opened with the question of monitoring with the people. And it's actually something we on crop diversity, which is a real actual issue now. So without further delay, let's hear Njido Kane, who will talk on Senegal biodiversity adaptation on drought-prone environment. Njido, are you with us?
Yes, I'm here. So just put enter.
You are with us, Njido? On the next one?
You are with us, Njido?
Yes. Can you? I will start.
Yes, please. Yeah.
I think you can just put the audio. It's easier. Okay, I will try live. Hopefully it will work. I'm very sorry, and I apologize to the organizer. I'm in Burkina Faso, and today we have a meeting here, so the connection is very poor. So I will just highlight here the biodiversity and how we use the crop variety of cultivated plant, cereal, edible legumes, and vegetables, and how also Senegal area where we used to feed the population in Senegal. So next slide, please. Senegal is subdivided into 6 agroecological areas, which shelter a rich variety of diversity, including forest ecosystem and coastal marine ecosystem, more than 700 kilometers. Around 8,000 species have been inventoried, including 174 cultivated varieties of crop composed of cereal, vegetables, and fruits to feed a galloping population. Christian, juste appuyez Enter à chaque fois. Appuyez Enter encore. Senegal adopts a national strategy and action plan to restore, conserve, exploit biodiversity for sustainable use, socioeconomic and environmental benefits for the population by 2030. His organization resolves around 4 strategic axes. First, reduce the pressure on biodiversity. Second, improve the knowledge on biodiversity and strengthen institutional capacity for the implementation of this strategy. Third, promote the inclusion of biodiversity in economic and social developmental policy. And last, promote the sustainable use of biodiversity and mechanisms for accessing biological resources for the benefit sharing of their exploitation. What is the status of plant genetic resource management? There is 2 conservation methods. In situ, which consists on farmers' field, protected area, classified forests, sacred woods, and agroforestry park. This type of conservation is very limited and lack of repertoire exists. The ex-situ conservation consists of gene bank, conservation stand, botanical garden, arboreta, herbaria, and also some park and forest, private wildlife area. This type of conservation unfortunately unconnect with conservation sites. Israel is a key actor of this national plan. Israel's goal is to contribute to the national plan on plant genetic resource management towards the establishment of so-called satellites, plant, forestry, microorganism, livestock resources. We have inventory more than 7,000 species that we conserve in gene bank in Israel, and also some plant we maintain by in vitro, and we also collect more than 5,000 strains of microorganisms. And these sites of satellites are spread across Israel center and laboratories. This rich and diverse agrobiodiversity is threatened by global challenges, anthropogenic and climatic. It is predicting a rise of temperature and a high variability of rainfall. West Africa is not spared. Model outputs indicate a downward trend in cereal production up to 30%. Upper panel show this trend in Senegal compared to other countries in West Africa, leading to economical losses. As you can see in lower panel, the production negative impact is amounting to millions of euros. Statistics show a diversification of culture. Millet is the first cultivated cereal, and peanut the first cultivated edible leguminous. For this reason, I have chosen to present you crop innovation, the tool, technologies, and proceedings we are using at Israel to develop new breeding product concept with our partners. Here are the new peanut varieties developed and registered that carry distinct traits and concepts: larger seed size, larger seed pod, high biomass, early maturity, and high yield. To develop those varieties, we explore and tap in the potential of white peanut species for breeding new varieties. Our global approach consists of implementing selection using molecular markers. We develop populations that we backcross, advancing this population using markers, and we detect some QTLs that are related to those traits. And then we validate those markers using the same population that we develop or other population where we pyramidize the QTLs alleles for breeding selection. And then we develop the new varieties that we test with some farmers and some other partners using participatory varietal selection. We also tap on the diversity of cultivated peanut. We collect more than 1,500 accessions from 9 African countries, some from East Africa, some other from West Africa. From that, we define a core collection that consists of 300 selected genotypes that are selected by breeders depending on the choice defined in their breeding product profile, and some other line also that we did using the core hunter software to extend the diversity. There was no geographical structuring when we analyzed this core collection, and we identified a structuration in subspecies of Arachis. Here are representing this division and species that we have developed using the core collection. And then we conduct some G by E by C studies, genotype, environment, and cultural practices. We know that most of the genotypes are influenced by environment. Here you can see in the panel on the top left how we evaluate these core varieties in our research station. And we have also some phenotype data of the pod weight and also the 100 seeds weight that help us distinguish different varieties and the diversity that we have in this core collection. Finally, we have conducted some GWAS analysis where we identify some QTLs genes that are associated with those traits. The other crops, pearl millet, is a staple food, drought tolerant and gluten-free. Pearl millets, among the other crops like maize, rice, and wheat, tolerate up to 42 degrees of temperature to complete recycles. Here you have on this picture on the left the domesticated pearl millet, and on the right the wild. I think in the presentation of Eve this morning you have the origin of the domestication explained. We have observed new genetic insight into pearl millet diversity as a result by the characterization of early maturing and late flowering landraces from Senegal. As shown in this picture, you have the late flowering millet, and on the right you have the early maturing millet. And this map represents all the points that we have collected to characterize this diversity. And we clearly see two structures of early and late flowering landraces of this diversity in Senegal. Morphological and genetic studies between early and late flowering landraces indicate 6 traits for early flowering varieties and 3 traits that are tightly linked with the late flowering varieties. And this also is linked to better performance of this variety in their environment. 6 of these variables are related to yield, biomass, and also flowering time, as shown here in the picture. We conducted some GWAS analysis, and we have identified 18 SNPs in the intergenic region and 24 SNPs in the coding region. All this information are used in our breeding product profile to identify some heterotic groups. We use the genetic diversity of these land races, but also we add some other land races coming from West and Central Africa, Niger and Senegal and Burkina Faso. To advance to the open pollinate varieties and hybrids development, we have identified some clusters that we make some cross using the half DLL strategy. Those pools have heterozygous and high quality of combining ability. Other processing we are using to develop also this new variety, we are using some genomic assisted selection that consists of making some interrogation and using some markers to develop those open pollinate varieties or hybrids. But most importantly, we are integrating farmers and gender selection in our breeding product profile. Here also in the picture in the top left, you see the woman choosing short pinnacle, while the men are using the long pinnacle depending on their preference. So these have to be integrated in our breeding product profile. A short anecdote here is the men are using the long pinnacle to ease the transportation from the field to their house, while the women are choosing the short pinnacle to reduce the labor when they are making a new product here shown in the picture. We are also considering in our breeding strategy what are the most vulnerable areas of pearl millet cultivation in Senegal and in West Africa under future climate change, and how the African monsoon will affect the agriculture in Africa. To assess the genetic diversity and vulnerability of pearl millet, we hypothesize that the use of pearl millet genotype diversity could help cope with the effect of climate change in West Africa and build resilient agriculture to climate change. We also assume that predicting increase in extreme weather events and uncertainty in rainfall pattern in the future is likely to affect agro-ecosystem and to threaten food security. To study the adaptation and vulnerability of climate of pearl millet, we use a wide diversity of pearl millet consisting of 173 traditional varieties, and we do the genotyping of those lines, and we use also the bioclim data models that consist of 17 variables. And we use as method, we use the Gaussian forecast modeling, which consists of machine learning regression tree-based algorithm, genomic environment relation prediction across space and through time, and landrace prediction under scenario of climate change. Some results indicate that late flowering varieties will be vulnerable to the northern part of the cultivation area, like here shown in the picture on the bottom right. Early flowering variety will be almost all vulnerable to climate change in the red box. Low variety linked with high diversity and cycle, mixing late, intermediate, and early varieties are here shown in latitude 12, in between the green and the red cycle. So the meaning of this result is that we can use this information to support decision making or for strategy, political strategy, or agricultural strategy development. When we use also the scenario that are described by the different RCP models, our result also indicates that we will not use biodiversity in pearl millet by 2050. However, the cultivation area will be really correlated with rainfall. So the next challenge will be really to define the hydrotypes that will be really high performing under G by E and C. C is the cultural practices. We are using statistical models to define these new hydrotypes based on G by E by C. We are also using some sustainable intensification practice like hydromix, which consists for us of using optimizing the services that are offered by the systems. So the message I would like to share with you here is there is a rich biodiversity we can tap to develop new varieties that are adapted to drought environment. We have to integrate also core farmers and marker features into our breeding product profile to make adoption easier and also make sure that what we are developing is what are needed and sustainable. We need also to identify the reservoir of diversity for adaptation to future climate change, to make those mitigation plan in place. We need also to assess the dynamic feature of crop to inform on the most vulnerable area of biodiversity evolution adaptation. And last, we have to integrate and associate the genotypes with the environment and the cultural practice to target new environment and define new hydrotypes and hydromix to increase productivity. So in conclusion, Senegal supports the commitment of the conservation of biodiversity through treaty and convention, and is right now through our research we support the commitment of Senegal to the conservation of biodiversity through treaty and convention. We also contribute to operationalize the vision to restore, conserve, and enhance resources by 2030. Community engagement is needed, and respect for ecosystem and beneficial, equitable, and sustainable use of resource system are to be well planned for sustainable provision and goods and services for the population. There is a need to integrate biodiversity issue into developmental policies, both public and local, supported by scientific proceeding, but also endogenous knowledge. And as biodiversity is large and rich, collaboration with all partners is needed also. So I will stop here by thanking all and giving credit to all the participants, credit to colleagues, students, and contributors. Thank you very much.
Thank you, Njido. You are with us?
Yes, I'm with you.
So, question? Yes. Do you have a...
So thank you, Guido, and so good that we finally were able to listen to you. And it was very interesting how women and men selected on different phenotypic traits in per millet, right? It was. So I was wondering if you could expand on that. Is it common with other, African cereals that selection by women and men would be different if the focus on different traits? And do you think that might have had any impact on the domestication and evolution of this species?
Yes, this is what we observe in pearl millet on other crops. It's something that is we are addressing in new studies, socioeconomical studies, to try to see if this is really linked with population and also use of these crops by the population. Now, to the question, is it related to the domestication? We do know that some traits, like the length of the pinnacle, was targeted by, always by humans to have a higher yield. So why women are using a shorter pinnacle is to reduce the labor in the house, because less people are working with the ladies in the house. So they select short pinnacle to reduce. I don't think it's related to the domestication, but rather to the new practice in the rural area.
Thank you, Guido. Another question, Jacques?
Yes, Jacques David from Supergro. You were mentioning the use of the open pollinated varieties for your outcrossing crops, and I found that it was really interesting to see that you try to develop that. But I have a very practical question. So for distributing the seeds to the farmers, what did you put in place in order to produce the local, I would say, female and local male population, and what are the structures that you think could be set in order to produce a hybrid between populations? So what about the seed system once you have described the genetics of this, what you have presented here?
Thank you very much. So the seed system right now is going through a lot of mutation. What we are doing right now at ISRA is to train seed producers, but also farmers, to produce this new varieties that we are provided to them. For the hybrids, we are the only one right now producing the seeds and giving that to the private sector for multiplication. What we have done so far is when we develop a new crop, we conduct some participatory varietal selection with farmers, and select those variety. We go through a process of certification of those seeds with one unit that we call DISM, who certifies the way that we produce the seeds. So really what we have put in place is really to be engaged in empowering farmers and seed multipliers and private sector, how they can multiply the seeds and keep them certified. The seed system, like I said, just going some mutation, and more people from the private sector are getting involved. So we hopefully that this will make this seed system more sustainable.
Thank you, Njido. Another question?
Yes, here. Thank you, Njido. It was very interesting to hear you talking about breeding breeding strategies in Senegal. But my question is that you spoke of breeding strategy applied on peanuts and pearl millet, and you just spoke on certification and seed system in Senegal. But my question is, how do you actually measure the adoption rate of those new varieties? Because we know that in Senegal, the seed systems are various, and people are using also farm seeds. There is a strong farm seed system. So how do you measure the innovation you propose and through the adoption rate of those new varieties? Thank you.
Yes, this is a question that come up quite often. For instance, what we have put in place is to conduct some survey to those that we provide the seeds, and to come back like 2 or 3 years later to try to see if they keep continuously using those varieties. And also what we have done this year is to recollect sample from their field, genotype them to make sure that it's the same varieties. This is more difficult like for pearl millet, but we did it for peanut and for cowpea, and we were able to certify that the seeds that we provide to the farmers are the same varieties that we find 2-3 years later. So plus that, we are conducting also some survey, like I say, and try to see what are the preference of farmers. Are they keeping continuously the use of the seeds after 2 years or after 5 years, or there is a drop? So this also need to be addressed more in depth.
Thank you, Njido. The time is going on. I would like to thank you for your presentation and your answers to the questions. I would like to thank also one more time, Victoria. Thank you very much. And Bruno, thank you very much for this session. It's time to go to the third session, the last one of the day. I would like to ask to Jacques and Mathieu to come to the stage. Thank you very much, everybody.
Okay, so hello everybody. So I'm Mathieu Thomas, I'm working at Arcad Institute, and here it's Jacques David, professor at Agro Institute. We are both geneticists and interested in crop diversity, evolution, and management. And we are pleased to introduce this final session that will help us broaden scientific approaches and methods to address the challenges of ongoing and future ecological, agroecological, and energy transition, and global change in general. Achieving this transition raises complexity challenges. The following talks are going to address 4 perspectives to tackle this complexity challenge. The first one, by Ruben Miller from University Juan Carlos in Madrid, Spain, as an evolutionary biologist, will make us aware of the interest of approaching the evolution of cultivated plants, from the point of view of their ecological traits. Then Salvatore Ceccarelli, thanks to his feedback on more than 30 years of participatory plant breeding at ICARDA and now at Biodiversity International, will present the challenges of considering participatory and evolutionary plant breeding involving farmers early in the innovation process. Then Carl Zimmer from Pennsylvania State University, as ecology and rural sociologist, will challenge us on the importance of reviving the links between plant breeding and food, nutrition, and health. Before giving the way to Thora Gain Herrera from ISESI University, Cali, in Colombia, who, as plant biologists, will discuss the challenge and limits of using new technologies, such as genome editing, to meet the challenge of sustainable feeding more and more humans in a changing and uncertain world. Thank you. And please, Ruben, you have the floor.
Where should I go? No.
Okay, so thanks to the organizers for inviting me to this nice event and the chairman for the nice introduction. I think we will be linking with the session of this morning. We will be talking about domestication and crop evolution. Again, but this time from the viewpoint of comparative ecology. So I would say like a naive and different viewpoint, perhaps. So first thing is to acknowledge that this is not individual work. This comes from collaboration from many people and institutions and around the world. And in particular, the results I'm going to show were contributed largely from my colleagues Nieves Martin, Pablo Garcia, and Alicia Gomez. So before getting to the very topic of crops and their traits and their evolution, I would like to think a little bit on what we assume as agricultural sustainability. So sustainability has many dimensions, right? And but we may agree that we cannot achieve sustainability if yields and the other services that we get from croplands are not sustained long term, right? And let's focus a little bit on one of the dimensions of sustainability, which is soils. So soils are key, right, for achieving sustainability. If we don't keep soil functions long term, then sustainability is not possible. And one particularly multifunctional and nice way of keeping soil functions is keeping or improving the organic matter status of the soil, right? And well, we may achieve better organic matter statuses in crop fields by management, right? By reducing tillage or rotating crops, diversifying crops, or keeping soil vegetation cover in the agricultural soils year-round. All those measures help to keep and improve the organic matter status of soils. But also we have to think about the plants themselves, the plants that we grow. We know that if we grow crops or varieties that have a deep and extensive root system, we will be putting lots of carbon into the soil, right? And if we grow species or varieties that have shallow roots, we will not be achieving that amount of carbon sequestration or organic matter build-up. And that applies also to many other ecosystem processes that go on in croplands. If we grow, let's say, canola instead of wheat, we will be feeding the community of pollinators that is around. Or if we grow soya instead of maize, we will be putting more nitrogen into the soil. And it's relevant to point out that it's not the identity of the species or the varieties that we grow, but their traits where it's relevant. right? Whether they're nitrogen fixers or not, whether they are big or small plants, or their leaves are very palatable to herbivores or not. That's what is relevant. But the thing is that the traits of the plants, as of any other organism, are not constant. They evolve, and this is the very topic of this talk and of the ones that were going on this morning, right? So if we think about crops, there are like two big events that are important to their evolution. One is the selection that we did of wild species for domestication, and the other one is how those wild species change during and after domestication, during their evolution and their cultivation. So let's first focus on this first selection. So did we pick species from the wild with particular profiles, or did we sample whatever was there? So if you ask this question to a general audience, people will start naming agricultural plants and will probably name the main cereals, the main Fabaceae, the main Solanaceae, those that are of course very relevant crop plants. But if you ask this same question to this audience, for instance, who is very experienced on agrobiodiversity, you will say, well, we grow and have domesticated or partially domesticated a very wide diversity of taxonomically different crops that also come from many regions around the world. So what is the actual picture when we try to put numbers into this? So do we use only a few specific clades of the flowering plants, or do we use all of them? One can answer this from many perspectives. This is just one possible answer to this. In this phylogenetic tree of the 1,000 plus food plants, you have in green and bluish colors, you have the branches and the tips of those food species that contribute the most to global food production. And if you go through the tree trying to pinpoint where they are, you can see that they are quite widespread across the phylogeny of this group of species, right? And if you use a different proxy for agricultural relevance, you at least the ones that we have used, you get a similar pattern. Or if you, for instance, map in the tree of flowering plants the abundance of domesticates or metrics like that, you also did that there is very little phylogenetic structure. So crops come from diverse phylogenetic lineages, right? And they also come from very diverse geographic and climatic regions within the places that we usually label as classical domestication centers, and also outside of them. Even if there's like a particular overabundance of crop origins within ecoregions that have like a seasonally dry period of the year. However, in spite of this like wide diversity or wide phylogenetic diversity and geographic diversity, we see that when we compare crops with the bulk of other wild plants, of botanical diversity in certain key traits, we find that they are not a random representation of all wild plants, and they have particular phenotypic profiles. Here, for instance, you can see that crops tend to be species with lots of nitrogen in their leaves, which means that they have a high photosynthetic performance, or they tend to be also large species with large seeds and large leaves and large organs when you compare them naively with the bulk of wild herbs or wild woody plants. In this case, you have here only herbaceous plants, which are the ones that we study. So we, from now onwards, I will only talk about herbaceous crops. And if you look below ground, it's almost is the same picture, but even more pronounced. The roots or the fine roots of the crops tend to be soft and tend to be thick as well. which has consequences for how they behave in relation to nutrient and water uptake, and how they relate with symbionts, root symbionts. Okay, so we have seen that you know, crops are very diverse, but still they have certain phenotypic profiles more than other phenotypic profiles. And what happened during their domestication, from the very step of the wild progenitors to the current varieties that we use? Did they change at all? Did they change their traits? And the short answer is of course yes, a lot. And we know a lot about this. And of course, they are so different. Their populations are very synchronous. The organs that were targeted by artificial selection have become generally larger, and they have lost the seed dispersal mechanisms and several other traits that tend to be common with domestication processes. But what happened to these kind of traits that we know are in direct link with ecosystem processes? Here you have like the generalized effect of domestication across 30 herbaceous crops on traits related to the size of the plants, to how aggressive they are as competitors for light, and to the resource use rate of leaves and fine roots. And what we find is that in general, during the course of domestication, plants have become generally larger and have become more aggressive competitors for light, but we don't find that they have become species that, you know, have a faster photosynthesis rates or uptake nutrients and water more rapidly than their wild progenitors. And here it goes in the same line that the last result I showed. So we also did like a wide screening of growth dynamics in crops compared to their wild progenitors in large set of accessions within crops. And we also find that in general, neither in general nor in particular for any given crop, domestication has promoted faster growth rates. Okay, so that might be the first take-home message. I think I will not summarize because we talked about it like just now. But the point is, well, there are certain phenotypic patterns associated with domestication that we know a lot about, and we know about the genetic architecture even, but there are also others, like the ones that I just mentioned. So do those changes have any impact on the ecological behavior of these plants that we use for agriculture? And again, I will exemplify with soil and soil functions, and in particular with the process of decomposition of plant residues, which, as you all know, is very important for nutrient incorporation into the soil. In the wild is so important. In croplands, it is also important sometimes. For a start, you are here a comparative view of how decomposable are crop residues when we compare them with the litters of, you know, plants that live in other ecosystems. And as you can see, we have crops as one could expect. We have crops that are decomposed very fast, but we have some diversity, right? Also, there are crops that are quite recalcitrant. So what was the effect of domestication per se on this process, on the decomposability of plant residues? So for addressing this question, we compared wild progenitors and domesticated accessions of a wide set of species, and this is what we found. Here on the x-axis, you have the decomposability of the residues of the wild progenitors, and on the y-axis you have the decomposability of their domesticated counterparts, so that each point in the graph is a different crop species, right? And if a point in the graph is in the upper triangle of that plot, it means that the decomposability of its residues is higher for the domesticated counterpart. So as you can easily appreciate, for the vast majority of crops, domestication has promoted litters that decompose fast, much faster than that of the wild progenitors. And that happens when we do experiments in soils that are rich in microbial life, which are, the organisms that are driving this process of decomposition. And that also happens when we do this same kind of experiments in microbially poor soils. So does this have any impact on the functioning of the soils themselves? And the answer is yes. Here is just an example. Here you can see like the amount of nitrates that you have in the soils after they are incubated with that litter and after that litter has decomposed on top of those soils. And as you can see, the enrichment in nitrates is much faster and higher in those soils that have been fed with domesticated litter rather than the litter of the wild progenitors. This is again like a summary across species, but the same pattern holds when you look at it species by species. So we may say that domestication has somehow sped up the nutrient cycling in croplands, whether for the good or for the bad. It's just fact. Would depend on contents and management objectives. So I had just exemplified with soil and this process of decomposition and nutrient availability in soils, but we don't have the time to extend on this. But here I pinpoint to recent publications where you can find examples of other impacts of domestication or evolution under cultivation for other aspects of the ecological behavior of plants. So, for instance, on how they interact with soil microorganisms and root symbionts, or how they behave in polycultures, or how they react and resist herbivory. Someone wants to take a picture or something, you have the references there. So just to finalize and going back to our initial scheme on sustainability, it is of course nice that we keep focusing efforts on how we manage agricultural fields and also on societal and political issues associated to sustainability. But I think that it is also important that we keep in mind that the plants that we grow themselves have impacts on dimensions of sustainability, and I'm 100% sure that everyone in this room is very well aware of that, because we are all from the agrobiodiversity field. But maybe outside this room is not that people don't have that in mind. And yeah, that is everything. Thank you so much.
Okay, good. So I said, thank you for this wonderful talk. It's amazing to see how domestication has modified so many things. So we started by looking at shapes, and you just indicate us that it has modified the complete ecological system. So I don't know if we already have a question, so please do not be shy. This talk was very informative. So if anybody has, I do. I have many actually. Maybe for the last one of the last pictures, you say that using the litter of the domesticate or the wild, you change completely the nitrogen cycle. And possibly you suggest that we have modified also the microbiota community in the soils. Did you try, or do you have any clue about the fact if you make a reciprocal transplantation for taking a domesticated litter into a wild environment and taking a wild litter into a crop as you did, maybe. Do we see something different, or is the microbiota also domesticated somewhere? Or?
Yeah, well, there's a bunch of of studies on. I don't know if with that specific design that you suggested with the litters, like doing reciprocal transplant between the litters, but we have done that with living plants, to know which are the, how a given plant impacts on the microbial community where it grows, and what is the feedback of that microbial community on whatever you grow afterwards, whether it is a domesticated plant or a wild plant or a different variety or whatever. And well, the general rule is that those feedbacks tend to be negative for plants, and that's why we do crop rotations, right? And but specifically, what we have found is that when we compare crops and wilds, is that crops tend to build up more of the antagonists, let's say nematodes, for instance, and less of the mutualists, let's say mycorrhizal fungi, for instance. So that if you grow them in the same soil and in the same field the following season, they will have a more negative feedback from the soil community, and that negative feedback is not that negative for the wild progenitors. So there is something in there that is has changed during domestication and makes them, those interrelationships with soil organisms not that beneficial as the wild progenitors.
Okay, so many things to inquire about, I guess. So a question?
Yeah, me again. Thank you very much, Ruben, for this presentation. I was wondering if you are now working with plant breeders in order to integrate the ecological consideration you've just introduced us today.
Mm-hmm. Well, I am not personally or in my group, but yeah, I don't know. We only managed to do this type of let's say basic research and comparisons and comparative ecology, but we don't have any active or prospective collaboration with breeders, which I guess it would be nice. Yeah.
So other questions? If not, I do have one. Okay, Hélène. Hélène, can you introduce yourself?
Hélène Fréville. Thanks, Ruben, for the talk. So you presented us some results with a relative growth rate where you didn't find any significant differences between wild and cultivated species. I was wondering if you had any hypothesis in mind when doing that, and if it's a surprising result, mostly in relation with the fact that for many traits like a specific leaf area and so on, you didn't find any significant shift. So how does it fit with the global, your global vision?
Mm-hmm. Yeah, it was not a surprise because we had like previous indirect evidence that was not that was not the case. Like crops have become generally faster growers during domestication. Probably the wild progenitors were already fast enough for agricultural use, and they have evolved other traits that are relevant. But it is a little bit, we have like many lines of discussion around that. And one of them is that, what it is general is that the defense compounds and the secondary compounds in general have decreased a lot with domestication, and that is quite general in most of the species. And from that one should predict that they have freed resources for growth, and they might have faster growth rates, and that is not the case. So that is one point where we are a little bit, well, then where do those all free resources go? I don't know. And yeah, and as regards adaptability to agricultural use, I think that the wild progenitors were already fast enough for agricultural use. There was no need to go farther into that direction.
Okay, we still have time for one or two questions. So if nobody, I have one which is burning my lips. You said that the domesticated seem to have more aggressive style or aggressive way of growing compared to the wild. But we imagine actually that the wild species were growing in a large community of other species, and we could have imagined that these conditions were even more stressful than in the field, but we have the reverse prediction. Actually, we could have predicted that it was more competitive into the wild than into the domesticated. Does it mean that the competition is harsher between the plant of the same species? That is to say, two domesticated are competing more harshly than a wild individual with others, with plants of other species?
Most likely. I mean, I think that this thing of crop plants being generally proficient competitors, and they invest a lot in stems and in height growth, and that was a problem that was solved for the main cereals in the Green Revolution, where they bred for shorter and more stout lines that did not invest that much in competitive traits. But I would say that a monospecific stand of cereals or any other crop plants is a fairly greater competitive environment than a interspecific stand of wild herbs. Of course, one has to measure that, but
ecologically speaking, it's a niche complementarity more in the
wild. For instance, there are other mechanisms like niche complementarity and phenological complementarity as well that may ease competition in wild stands more easily than in monospecific.
Okay. Darwin told us that the competition was harsher with your king or your more resembling. Okay, I think that if we don't have any more question, any question there? Mathieu, you want to have a question? So thank you again, Ruben. It was great. And so we are going to go to the second talk. I guess, Salvatore, are you in online?
Yes, I am. Can you hear me? Hello? Can you hear me?
Yes, we hear you, but we are waiting for your presentation. 2 seconds for starting. Yes.
Hello?
So, yeah. You are, yeah.
Can you see my
Perfect.
first slide?
It's okay. Perfect. Thank you.
May I start?
Please, Salvatore, you can start.
Okay. Well, thank you very much for the introduction. I'm actually not working for Biodiversity International. I'm simply a consultant on a large project that will be the last part of my presentation. My presentation is fairly general compared with the previous one. I would just like to show you some genetic material which can fit very well into an agroecological system. And these are evolutionary populations. Let me start very generally with the global problems we are all aware of, such as climate change, water, the problem of food and the malnutrition, the use of pesticides, and then also biodiversity. Just to make the point that at the center of all major problems, in one way or another, there is seed. And among these problems, I would like to pick up for a moment climate change, just to make the point of that this is a phenomenon of an extraordinary complexity, because although we always thought about, we always think in terms of temperature and rainfall, actually with the change of temperature and rainfall, now we have a change in the spectrum of diseases, insects, including pollinators, and weeds. And therefore, for example, plant breeding for resilience or adaptation to climate change is a very difficult task, because this is going to happen in different ways in different parts of the planet. In the case of biodiversity, I would like to point out that there is a big contradiction in science. The major journals are always telling us that biodiversity is terribly important for food security and hence for resilience. And again, we are connected with climate change, but also for our health. And on the other hand, the science which is producing our food, as everybody knows, has gone for the last, 78 years, almost consistently towards uniformity. And this is the most common agricultural landscape which surrounds us. And very often we are told that this is needed to feed the world. And now we know that is not true. And that is not true comes from this report on food security and nutrition in the world, which is published by FAO, UNICEF, IFAD, World Health Organization, and the World Food Programme every year. The 2021 is not yet out, which shows that after years of slow but constant decline, the number of undernourished people in the last 4 or 5 years started to climb again. So it's not able to feed the world, but it's also endangering our health from 2 points of view: the use of chemicals and the type of food it produces. The type of food behind all these apparent diversity, the reality is that 60% of our calories are coming from just 3 crops: wheat, rice, and corn, which are not the most nutritious crops and which are using 50% of all water used in agriculture. It is also endangering our planet because it contributes for nearly 24%, if we are not considering part of the 14% due to the transport. Everybody is aware of the large waste that we do. Just walk around a supermarket and you will find something like this. But now what is coming out is that the domestic waste is on the increase. In the case of Italy, it's been estimated at 57 kilograms per person per year, and it contributes of another 8 to 10% emission. Now, what is the solution? What is interesting is that in the same report that I was quoting before, there is not a single word about biotechnology as a possible solution. But there is what is mentioned very often is agroecology, ecological food, and organic products. So one obvious way, one obvious possible way to try to revert this trend is to return to diversity. And this, as some may know, we've been doing by practicing participatory plant breeding, which is nothing else than transferring the breeding trials into farmers' fields, letting the farmers express their opinion, and this invariably brings to the selection of different material in different places due to different soil conditions, different cultural reasons, different uses of the crops. The idea of participatory plant breeding is started in the 80s with this classical paper, and it appears now as an interesting model of plant breeding to address three concepts. One is that of client profile, which is basically breeding for whom. The second is the product profile, in other words, breeding for what. And the third is the environment profile, breeding for where. Now, some of these concepts are part of the private breeding, but they find very hard to find their way in the public breeding. In addition, participatory plant breeding it can exploit rather than avoid genotype by environment interaction. It maintains the circular fashion of a formal breeding program. The institution generates a genetic diversity, which is immediately deployed in farmers' fields. Farmers, in an inclusive manner, do the selection, express their opinion. They select the best varieties for their own place. These varieties are coming back to the institution for another cycle of recombination and selection. And at the same time, farmers become seed producers. The model has had a number of successes and recognition. Has been recognized as being able to generate spatial and temporal agrobiodiversity, to empower farmers, to generate resilience, to produce adapted material, and to increase breeding efficiency. We have been practicing participatory plant breeding in a number of countries and with a number of crops, but institutionally, the method has not been, with few exceptions, has not been accepted. And we argue that this is probably due to the difficulties of several breeders to share the merit of selecting a new variety with others. So during the last few years when we were in Syria, we thought that evolutionary population could be, in addition of being an agroecological solution, may also get around this problem of institutional acceptance. We are talking of a very old science. The first paper on evolutionary breeding, even though with different names, dates back to 1929 with this paper by Harry Arlan and Mary Martini. But it took this name of evolutionary plant breeding in 1956 with this American agronomist who actually published a recipe on how to build an evolutionary population, getting varieties from very different places so that they were very, very different, cross them in all possible combination, bulk the progeny, and let this big mix with a lot of genetic diversity to natural selection in the actual environments where where a new variety was needed. And the same Sanderson was able to show shortly after that actually this population do evolve, and the yield is increasing steadily. The straight line that you see, the horizontal line, is a very common variety at that time, and you see that all these different populations manage to either to get very close or even to go above the yield level of this popular variety. The dots that you see on this graph is actually another aspect of the evolutionary population which consists in using them as a reserve of diversity from which the breeder can actually select. As I mentioned earlier, this was more or less the last piece of work that we did. We left Syria in 2011. So in 2008, we made this population of barley. This was the crop on which we were working by mixing the same number of seed of 1,600 F2s And the year after, our colleagues working on durum wheat and bread wheat, they gave us their material and we did the same. But these populations were sent to countries where we were already practicing participatory plant breeding, because those farmers were already very skilled in managing diversity. These populations eventually came back to Italy within the framework of a European project, and they were introduced by the Italian Association of Organic Agriculture in the framework of that European project. And this is how one of these evolutionary populations looks like, a very different look from a modern variety, being made of thousands of genetically different plants. As I mentioned before, there is quite a lot of science, quite a lot of scientific research on these populations. And basically what happens is that even in crops such as barley, wheat, rice, there is always this 3, 4, 5% of natural crosses, which followed by recombination and coupled with natural selection, make in such a way that this evolutionary population evolved. In other words, the seed that you harvest is not, genetically speaking, exactly the same that the seed that you planted. And therefore, as a biological product, they're just the opposite of the product of biotechnological tools, which they still maintain the uniformity, which is the prerogative of modern varieties. And just to give you an example of the ability of these populations to evolve, if you look at this slide, you may hardly believe that these two fields actually are coming from seed, which 10 years ago was in the same bag and was simply given to two different farmers, one in Sicily and one in Tuscany. They planted this material for 10 years. And when we bought the seed from the two farmers and we planted side by side, this is what we found. They were completely different. The Sicilian population has this light green color, is shorter, more resistant to drought, more resistant to lodging. The population from Tuscany has a higher yield potential, but it tends to lodge when it's planted on fertile soil. But the point I would like to make is that because of this diversity, and again, there is quite an incredible amount of literature supporting this, they control pests much better than uniform varieties for obvious reasons. A disease or an insect has to find a way to find the resistant plant. Ecological science tells us that in this big mixture, natural selection favors tall plants, which of course in modern agriculture is a disadvantage, but in organic agriculture is a big advantage, because this very often is not associated to lodging and has the beneficial effect of controlling weeds in an almost perfect way. And therefore, this population basically can be easily grown without pesticides, and therefore they are the ideal biological material for an agroecological system. And that's why I mentioned the role of agriculture, because of the reduced use of pesticides. They mitigate the effect of agriculture on climate change. Just to show you the ability of this material to control weeds, these are some fields in different parts of Italy which did not receive any chemical at all, and you can hardly see a weed. And in fact, one of the that's interesting. So sometimes it doesn't work as a. And in fact, if you look in this field right below, you can find some weeds which are hardly able to see some field. And usually when we bring visitors to see this field, comparing with the field in the background, the first question that they ask us, how did you control weeds? Because they don't see the track of the tractor. We spray the herbicide, and yet there are no weeds. So basically, they are an interesting genetic material because they combine both a mitigation strategy to cope with climate change, but also with an adaptation strategy, because as they evolve, they manage to cope with that complexity of climate change that I was talking about before. And last but not least, the farmers are becoming interested for biological reasons rather than for ideological reasons to keep their seed, because there cannot be better seed.
Salvatore, you
Yes.
have 3 minutes left. I'm sorry.
Sorry?
You do have 3 minutes left. Sorry.
Yeah, I finished. The economic side of the story is that they have generated an interesting supply chain with all sorts of different products, being bread and pasta are very common in Italy. Very often they call it miscuglio di Aleppo, remembering the place where they're coming from. Another country where the idea spread very easily is Iran. And in fact, Iran is one of the 6 countries in this project managed by Biodiversity International in Jordan, Uganda, Nepal, there's Bhutan, Iran, and Ethiopia. And we have now, in this part of Italy where I live, this small project where we actually bring visitors and common citizens to see what is behind a loaf of bread and a dish of pasta to make this product more acceptable to a wide range of customers. Thank you.
Thank you very much. I hope I did not disturb you too much because you still have time left. So I don't know if I don't know if we already have one question in the room. Yes, we do.
A question from a distance.
It's okay.
Can you introduce yourself?
Just a very practical question. In your method of evolutionary breeding, you say you start from an F1 between two wheat population. Practically, how do you make an F1 between two population of autogamic plant? I don't understand the practical aspect.
Sorry, probably, can you hear me?
Yes, we do.
Yeah. Sorry, probably I did not make myself clear. Actually, the population that we made, they came from a wide range of F1. This was basically our breeding program at Arcad. In that particular year, we made 1,600 new F1. Each F1 was simply the cross between 2 parents, which were pure lines. So what we did, because we did not have enough seed of the F1, we mixed the corresponding F2. When we were preparing our international nurseries, we had quite a lot of remnant seed, and we used the remnant seed to make the population. And the durum wheat and the bread wheat, they're coming from the same. So it's not an F1 between 2 populations. It's several F1, each one from 2 pure lines, 2 varieties or 2 breeding lines, which were then mixed together.
Yeah, Carl, go ahead, and then a question in the room. Carl, you want to ask a question to Salvatore?
No, I think it's been covered. Thank you.
Okay, thank you.
No, it's just a quick answer to the previous question. I think it was related to the biology of the plant. And what you do is just to sterilize the panicle. I mean, you sterilize the pollen, and then even if it's autogamous, you can cross them and obtain F1s.
Yeah, sure. I mean, this is what we've done for 30 years. We had what in the jargon of breeders is called the crossing block, which is a separate nursery where we have a number of standard parents which stay there for years because they bring in genes for resistance to different diseases. Being an international breeding program, we had also genes for resistance to diseases which were important for some of the countries we were serving. And then there were newcomers. For example, we in the case of barley, we did an extensive work with the wild progenitor of barley, Hordeum spontaneum, because we found in the early days, this was in 1987 when we had a terrible drought in Syria, that in a huge nursery, the only survivors were a few lines of Hordeum spontaneum. So they became immediately the best candidate to go into the crossing block. And in fact, most of the best lines that we had for the dry areas were coming from this material. Of course, they were limited. And then, as you said, because the floret, or in the case of barley, the same in wheat and rice, you need to emasculate the plants that will act as female. And then there are different techniques to bring the pollen of the male plant into the spike, which has been emasculated, and therefore it becomes only a female. And usually you do this work in a plastic house, because if you do it outside, there is always the danger of pollen flowing around, and then while you think that you are emasculating, then you get some uncontrolled.
Yes. Salvatore, we do have another question in the room there. Lorraine?
Yes. Hi, Salvatore. This is Lorraine Gay speaking. Thank you very much for your talk. I had a question regarding the populations that were selected in Sicily and Tuscany. I was wondering whether you tried to measure the yield in each environment and to do sort of a reciprocal transplant of these populations. And if you did, what was the result?
No, we didn't. We received data from the two respective farmers, which of course are not comparable. At the moment, we have an experiment in northern Italy in a farm, and I only got the data, the agronomic data, and not yet the yield. What we have done, though, which might be of interest, is that in discussing with farmers looking at these fields that I show you, the idea came from the farmers that why do not mix the two? And in fact, we did it last year. I mean, the cropping season just finished, and we found that the Sicilian one was standing. The Tuscany one went down because there were some late rains, but the mix of the two was very good, and the yield of the you have to accept that these are yield coming from farmers' fields, so are not are certainly not publishable. The yield of the Tuscany and the Tuscany plus mixed with the Sicilian is certainly higher than the Sicilian. But we have not done the transplanting because these farmers have generated their own supply chain. For example, the Sicilian is selling all his grain to a miller, which is supplying most of the pizza places. So if you come to Italy, probably you will eat pizza coming from these populations.
Thank you. Do we have another question? So I do have one. maybe you know that in France we have carried out this gestion dynamic management of bread wheats. And actually what we have done was to make evolving evolutionary populations, but our results were a bit contrasted to what you explained. That is to say that we found that the yield... Oh, sorry, there is one question from Bruno. Okay, so I stopped my question. And he said, so Bruno, I will, if we've got time, but I prefer to give priority to the question of the people. So forget my question, and we do have a question from Bruno Fadi at INRA. He asked, what about the legal side to this? Only registered varieties can be commercialized. There's a question, no?
Good question. Well, in 2014, the European Commission started what they call a temporary experiment, and within that framework, a temporary experiment which ended in February this year. And within that temporary experiment, it was possible to register populations of bread wheat, durum wheat, maize, oats, and barley. Now, with the new European regulation on organic agriculture, that idea of legalizing the commercialization of seed or population has been extended to all crops grown under organic conditions. The way of doing that seems to be simplified compared with the temporary experiment, in the sense that the only thing that the farmers have to show now is not necessarily that the field of, let's say, an evolutionary population of bread wheat doesn't have any barley or any durum wheat, but it only to show that there are no diseases going around with the seed that is selling. So from this point of view, is a revolution in the seed policy, which is, as for now, is limited to the organic world. But if it is successful in terms of increasing resilience and adaptation to climate change, it may well be extended to all agricultural systems.
Okay, so yes, I think that this regulation will change a lot of things concerning possibility to grow for the farmers this heterogeneous population. So I think that we don't have any question left. So we will thank you again, Salvatore, and we will move to
Thank you for inviting me.
Yeah, it was a pleasure to hear you and to have your message, your very important message about diversity into the fields and into the varieties. So thank you again. So next, it's Carl Zimmerer. Are you ready? Can you take the screen? Maybe Salvatore, maybe you should have to leave it, to unleash it. Say okay.
Yeah.
Okay, so that's good. Perfect. Thank you. So Carl, when it's up to you.
Great. Can you see the screen okay? And can you hear me okay?
Perfectly. Clear and sound.
Oh, great. Well, I just would like to begin by thanking you for the invitation to join today's meeting and for the introduction, and also just to say how much I've been enjoying each of the talks this afternoon or the morning where I am, and they're just interesting and important work. And my invitation and focus is to think about crop diversity in food, to talk about recent research that I've been doing in this area, and to focus on future transitions. So I would like to have a kind of central focus on the crop diversity food interactions, but have a larger context of future transitions. And one of the the transitions that I'll be talking about, which is shown on this first slide, is the role of urbanization and what urbanization means for the crop diversity food nexus. And I think that traditionally and conventionally, we've always seen urbanization as the enemy of crop diversity. But what some groups that I'm working in are really interested in promoting is how urbanization can be and already is in some ways positively associated with crop diversity food interaction. So that's a bit of the larger context here. So the overall framework that's being used here is a 4-theme framework. This is very much a kind of transitions framework. It's something that colleagues and groups and I have been doing for a few years now, and it's kind of taking what's over on the left side of the screen, the agroecology and genetic resources focus of crop diversity and agrobiodiversity research, and seeing it in an interactive perspective with food security, nutrition, health issues, global change kinds of processes, climate change, food systems change, etc., and also governance influences. And so, you know, I think this is a approach that has a lot of opportunities associated with it, because agroecology and crop diversity work will always remain focused on plant breeding and agroecology and genetic resources, but there are these increasing kinds of initiatives and pressures and opportunities and challenges that come from the future transitions. That's part of the title for this session. So this is kind of where I'm coming from in thinking about these themes, and I'll be talking a lot about a couple of case studies in the countries of Peru and Bolivia, especially a Peru project. The photos are on the bottom here. This is a very interdisciplinary project with nutritionists, genetic resource and agroecology specialists, as well as global change and social ecological systems kinds of work that's part of it. And I'll really focus my comments and some slides on the main axis there between crop diversity, agrobiodiversity on the left, and food interactions with food over on the right, because this has become a really central focus in this work. But I will refer to urbanization, climate change, some of the global change processes too. And I need to acknowledge and credit and recognize that this is a large collaboration with many really great and important colleagues who are working on this project. And what I'll talk about today is the data from basically 2016 to 2019 first phase of this project. Very extensive, but we're actually going to be able to, we have some new funding and we'll continue it and be able to do comparative studies both after or at a different phase of the COVID pandemic and compare them to this sort of pre-pandemic situation, and that's pretty interesting. Anyway, here's a little bit about the study area where the data is coming from. This is an ecotone of the Andes Mountains with the Amazon basin, mega biodiversity area in both quote unquote natural biodiversity as well as crop diversity and agrobiodiversity. And it's about 200 miles from the capital city of Lima, but very integrated in terms of people migrating and market sales of everything that people grow, or much of what they grow. So I'll talk more about that. There are 3 areas in this study, or sub areas they're called. There's a peri-urban area. There's kind of a classic Andean area, which is the purple one. And then there's a highly commercialized commercial potato growing area over on the right that supplies the Lima market with the really popular Papa Amarilla yellow potato that's served with ceviche. So we have like really different contexts. There's a variety of methods. I'll talk a little bit about the sampling and the focus groups, but really focus on some of the survey and nutrition data to get at this question of interactions between crop diversity and food, and some ideas for what in my own research I plan to have as an upcoming next phase. So biodiversity indices, these are, you all probably already know these. These are widespread and common in the literature, although they're often not reported and described, in my opinion, nearly as carefully as they can and should be. We continue to need to benefit from really rigorous, careful use of these statistics and which form of the statistics, etc. In this case, we actually created estimates with all 6 forms of these different measures. They're geocoded field sites. So there's a kind of spatial data analysis that is already being used and has more possibilities. Some of the initial modeling of the crop diversity outcomes shows a combination of various significant factors. The ones that are in bold and underlined are the ones I want to highlight. And I'm sorry, this was given in Spanish a few days ago, but the alimentación producida localmente, the locally produced food, is an important predictor, as are traditional foods. It's interesting too that some of the agroecology predictors are significant. Legume rotations, thinking about these kind of functional meanings of crop diversity is really important. But it's this is, to the best of my knowledge, this is the first work in which food has served as a kind of functioned as an independent variable, looking at crop diversity outcomes as dependent variable. Usually it's the other way around, and I'll talk about the other way, but as I kind of had wanted to say at the beginning, this main axis between diversity and food is a two-way kind of directionality of influences. And so these local foods and traditional foods are significantly associated with diversity. The overall model results are similar and better than some that, or most that are in the literature right now. I think one of the interesting things, and one of my next steps, will be to think about different ways of categorizing traditional foods to potentially improve the explanatory value of this model. Right now we have a extremely kind of strict and narrow definition, so it's only the sort of pre-Columbian foods that are traditional, and obviously many of the post 1500s food plants are really integrated into traditional diets. So I think that's an interesting kind of opportunity. Now, here's the work where it's real, the arrow's going in the other direction in terms of crop diversity influences on food and nutritional outcomes. And this has benefited a lot from these really elaborate data sets that we have using what are called 24-hour recall techniques. So we're able to derive estimates of dietary diversity and a minimum dietary diversity estimate, species richness, and then these micronutrient-based adequacy estimates. And there's a lot to be shared here, but I think probably the important take-homes are that crop diversity at the level of species richness is significantly associated with these favorable nutritional outcomes in terms of MDD, minimum dietary diversity, micronutrients, and that female-headed households tend to have a significantly higher chance to show this association between crop diversity and nutritional quality. So it doesn't mean that like all women-headed households are doing this, but many of them are. And we're another interesting part of next research is to kind of look at heterogeneity in that population. So another important explanatory value. So these should have said this. Sorry. The IRR. These are multivariate statistical approaches. Values greater than 1 represent positive associations. So here it's showing how garden cultivation is positively associated with various kinds of nutritional outcomes. So this is self-growing, and this needs to be put in the context of very, very real environments, food environments and situations throughout Peru and Bolivia, but really Latin America as well as Africa, Asia, where maybe it's 1/2 of people's food that's being self-produced, or 1/3. Here it's about 1/3. Obviously, in other that's going to vary a lot, but it's almost always a combination of market and self-production. So one of the key points for me is recognizing how the self-production, the agrobiodiversity that I've been talking about, is in a sense making a difference at the margin. It's not that they're self-producing all their food, it's only roughly 1/3, but it really is creating these nutritional impacts evident in this slide. This is the previous slide. Again, the values greater than 1 being the significant positive associations where there are asterisks. Let me keep going with some of this work. Yeah, so basically we have some localized differences among the areas. Values less than 1 indicating that they are less diverse than the reference area. The variation here was a lot less than we had expected. In fact, the peri-urban area, which is located just a few kilometers outside of this medium-sized city, it's called Huánuco. So it's a city of a few 100,000 people. The peri-urban area, it's significantly less, but by a pretty minor degree. So this is one of the first things that in my work has started to make me interested in the role of urbanization and urban spaces that I'll transition to in a minute. It's important to recognize in this context, in many of the contexts where we work with crop diversity and food, that these are food insecure context, what we see in hopefully it's visible, numbers 1 through 8 are just the different kinds of questions that are asked in order to create a food security estimate. So these are different, these are standardized questions. It's a standardized format, and you can really see that the levels of uncertainty and worrying about food availability are really extensive and are part of this backdrop. When we actually delve into the data, it's the most food insecure people have less dietary diversity and less micronutrient availability. They're self-producing less food. This is probably the this is the most marginal group, whereas the least food insecure, the relatively most food secure, have positive values in all of these respects. So this is for much of the world, for the marginally food insecure people that we work with on issues of crop diversity and food security, this is a really important finding, and it's really important, in my opinion, to probably begin to be a lot more explicit and incorporating into research when and if possible these issues of food and nutrition security. So let me take a couple of minutes and talk about the background and what I'm thinking for some of the next steps in this work.
Excuse me. Excuse me, Carl. There is 3 minutes to go.
Okay, thanks. I'll do this quickly. So just to say that these dynamic transitions are really important, and the climate change situation is one where basically potatoes are becoming less common and maize is becoming more common because of upslope shifts. And so what does that mean for food and nutrition is an important question. Transitions to part-time farming, which can be very positive. This is the really important work on migration being a positive predictor of crop diversity. So again, recognizing that these future transitions, while they're more complex, they're very interactive, as we've been talking about. It's not just one of them. Often 2 or 3 or all 4 are going on at the same time. And most recently, it's the urbanization transition. So in this sketch of proposed work, I'll be looking at this same data that I've been talking about, but with additional focus on the role of foods, different concepts of what constitutes traditional, cooked and uncooked. I'm really interested in the role of recipes. Nutritionists have a very break it down into the nutrient composition approach, whereas I think a recipe level analysis of this data is extremely important and a very promising next step spatially, and then also to really think about it temporally. So combine what's happening in the rural areas with what's happening with the same families and their other family members who are now living in cities. This is the kind of data that we're looking at. So there are several thousand rows of this. The second row is the recipe data, 3 recipes here. You can break each of these ingredients down into the nutritional components. And also, of course, there's the crop diversity data for each of the households. So this is an extreme, there are about 1,200 recipes in this data set among the 300 women that participated in the survey. This is just my last slide, which is to say that it's really thinking in this broad picture about the food and crop diversity axis in the context of all the the future transitions, including urbanization, and recognizing that there are many, many challenges, but hopefully really positive, important opportunities as part of this also. Thank you very much.
I'm looking, there is no question. So the time that people are thinking, I was wondering, Carl, if you could see a relationship between the labor-intensive way of preparing the products and the level of diversity. In other words, is the size of the household important in the number of species that the household is cultivating or using or buying or selling?
Mm-hmm. Yeah, I mean, we did test the labor variable here, and it didn't prove to be significant in this analysis, which in some ways is surprising, because in several of the other models that I've worked on and that many people have worked on, it is important. I think one of the interesting things, this is a very, very much a part-time farming situation, that the average farm size is about 1/3 of a hectare. That's very small, of course, and it means it's associated with people doing a lot of non-farm and off-farm work, part-time farming. And what they've developed are a lot of labor-saving approaches to crop diversity farming, which I'm absolutely fascinated by. I think it's really important. So they have an approach to polycultures, for example, that is much less labor intensive than other kinds of field-based cultivation. So they managed to get a lot of crop diversity while using just small and intermediate levels of labor. So I think that's what's behind labor not having turned out significant in the analysis. Thanks for that question.
Yes. So there is no question still in the room. So I go on this question of labor. Can you characterize every species that people use by the quantity of labor to get, I don't know, maybe 1 calorie or the sufficient amount of matter for a recipe or for a meal? Is it possible to detect if people are, when they live, for example, in urban areas, or if young people are leaving the household, what are the first species that are get rid of?
Mm-hmm. So I think that's a great question. At least for the field crops, we can definitely come up with a labor cost per unit of yield or production, because it's in this data set. And that's pretty extensive, and that's a really interesting idea. Thank you for mentioning that. In terms of which crop they give up first, this has been a focus of mine for more than 40 years. And in these contexts, it's very clear that there's one crop that's now on the verge of extinction, which is the one that they most commonly give up. And it's Solanum foraja, which is a fast maturing potato that has a growing cycle of less than 3 months, and it's particularly non-dormant. And so basically it evolved historically in cloud forest areas where they would repeatedly grow it 3 or 4 times a year. It was essential for off-season food inputs. But imagine the amount of labor that is required in this plant that doesn't have seed dormancy and needs to be replanted. So in this new paper in Cochabamba in Bolivia, we document it as the first extinction in this area. And that's my colleague in Cajamarca in Peru, who's come out with a book where they're finding the same thing throughout Peru. And that's entirely a labor savings driven issue that's driven the extinction of, or near extinct, it's not extinct, the near extinction of Solanum pareja. Thank you for mentioning that.
Oh, there is. You've seen somebody? Christian?
Thank you, Carl. It's a real pleasure to see you. I have a question concerning we all of us experiment the difference between what people live and how we describe what they do from outside or from inside. And my question is concerning the way of defining biodiversity or agrobiodiversity. Using index as Shannon, and many tools that only exist in our side, Western side, concerning society where there are not Shannon index to manage the biodiversity that they use to eat, to share and to interact with. My question is this one, according to you, there is a correspondence between the diversity as we try to define from our side and the diversity they manage daily in their side.
Yeah, hey, thank you very much, Christian, and it's very nice to see you and to see everyone and to hear this question. Yeah, I think there are a couple of answers. One is like an obsession that I've had for a long time, which is how people's own views of diversity are grouped spatially in the landscape. And I think this is really important. Maybe it's more important in the Andes Mountains because the landscapes are so varied, but I really think it exists everywhere in different ways. So spatially, there are categories and concepts for grouping diversity, which I think are among the most important ones that are not part of our sort of Western biodiversity science, because they're really categories and concepts of space. I wrote an article once, I called it ethno-landscape ecology. So the idea of people's own concepts of agrobiodiverse spaces. And I think that that's a really important for them. I'd say that where I'm going with this new work is, in a way, it's similar. I like to think it's not nearly. This is a big compliment to you and the GEC framework. But I kind of imagine whether food can become another form of C in our kind of thinking about this. And so honestly, that's my interest in the recipes. I wonder if, it's food dishes, right? Each one of those has a local name. It has a local custom in terms of what ingredients are recognized and are socially shared in terms of knowing about, and recipes are shared and they're passed down in families. And so I think that that's another area of grouping of biodiversity and a cultural sense that's completely missing from Western biodiversity science. And that's what's drawing my new interest to this focus on the role of recipes.
Thank you, Carl. It's fascinating to see how complex are the culture around the food and the species. It's just fascinating, and especially in Southern America, such just with the solar, it's fascinating. So thank you very much. I think we are going to listen to Tora now, and you have seen that this this symposium, we started from Neolithic this morning, and now we will be in full 21st century and maybe beyond. So Tora will speak about genome editing as a new source of diversity and possibly the risks that are associated and maybe the benefits.
Yes.
Good afternoon. Thank you very much for the invitation to participate in this reunion, this meeting today. I will be presenting some of the aspects of my experience during the last month of this year as an invited researcher in the Montpellier Advanced Institute of Knowledge for Transitions, Muse University, with the support of CIRAD, the UMR Arcad. And this briefly, what is important about this experience was an invitation for scientists to get out of our over-specialization bubbles and analyze the potential contribution of our resource in the light of many other options. And getting out of this bubble and start looking at our proposals not as the only solution, as only one solution, but as one of the many different solutions available.
I'm sorry to interrupt you. To fix this.
Okay. Okay, well, just an invitation not to look at our potential contribution as the only ones, but to analyze them critically in the light of the plurality of options that are available when we talk about agricultural transition. And happily, this participation in this meeting this afternoon with these 3 previous talks just confirm this need of getting out of our narrow view as scientists and integrating all this plurality of options and deciding in the long term which of them will be better when we talk about sustainability. No? It's not moving?
C'est bon là? Non plus. Non. C'est pas le bon écran. Hein. Christophe, si tu m'entends. Il arrive. It's coming. Ça marche.
Peut-être parce que c'est un PDF. C'est pas PowerPoint, c'est PDF. So with this view, talking about genome editing is very important in this moment. Maybe you are aware of the discussion that is taking place just now. Here in France, French are very engaged in discussing genetic technologies, maybe sometimes reactive. And this discussion happened by pause, maybe a spasmic discussion. And now we are in one of these very important moments. Since 2018, we are discussing how we are going to consider this genetic technology, especially gene editing, and how, when, and for what we are going to implement genome editing when talking about agricultural transition. And this is just an update of what is happening now. In 2018, there was a ruling by the European Court deciding that genome editing can be classified as a transgenic technology similar to GNO, which has been a very long time considered in a very regulated way. But this was contested by scientific communities, and then was an invitation to a targeted consultation by the European Union. We decided that this regulation is not adequate for considering genome editing and other genetic technologies. And now, in different countries of the European Union, in England, in the US, around the world, we are having this kind of discussion how we want to regulate this, because this is very, very closely related and how we are going to use it and for what. Why genome editing? Well, just a reminder, many of you know what is genome editing. This is an array of technologies that allow us to intentionally and in a very precise way change genetic information, knocking out genes or introducing genes for expressing desirable traits. And when we talk about this, we have many different technologies that show difference in the level of the changes, in the kind of reparation mechanisms that take place after we do the changes, and whether they imply insertion of DNA from foreign species or from related species. So all this classification, all this variation in the type of technologies are very linked to the regulations. We are going to frame them, but also with the applications and with the potential risks we are going to link with these different applications. From these technologies, maybe the better known is CRISPR, and it's the preferred one for its precision, its cost, its rapidity. And we are now taking of other kind of application, gene drive, which are very, very related with the subject we are talking today, biodiversity and how we can manage diversity, and which is a very hot topic when we talk about implementing these genome editing technologies. And here is a condensed presentation how these different technologies can be considered in the light of regulation. I'm sorry for insisting a lot about regulation, but regulation is what today allows us to decide whether or not to use these technologies. And worldwide, there are two different frameworks, two different approaches to consider this analysis of viability of the different technologies and its application. One is based on process, is to say, for example, as the European Court said, we just look at the process and for us everything is GNO, or we are going to analyze the situation case by case, looking at the different characteristics we want to manipulate, what is called the product-based approach. And which is happening now is that scientific communities around the world are pressing for the product-based approach, and this is one of the reasons and give the emphasis to very product-based examples in my presentation. So this the diagram is quite complex, but what it's showing us is different technologies, how stringent we will be regulating them depending of the different changes they are implying, the applications, the environmental risks, etc., etc. One of the questions is, and especially after listening all the presentations today, is why using gene editing when we talk about diversity, when we talk about trait diversity, when we talk about genetic diversity. And there are different frames that justify using genetic technologies and genome editing in an agricultural context. Some of them are associated with the genetic bottlenecks caused for the domestication processes. So genomic technologies, genome editing appears as a solution to solve the problems caused for domestication of crops and from the dominant system of production, the intensive production system. So there are justifications narratives relating with food security crisis. We need to feed the world population, and gene editing offers a faster way to do this, to have more nutritional crop with higher yields, more resilient crops to may face to climate change, or to migrate our intensive agricultural system to an intensive, more sustainable agricultural system. We can contest this different frame. Part of my exercise being here in Montpellier was to analyze these different frames and try to critically contest them. one of the characteristics of the different frame is that it's monoreference. We are valorizing genomic technologies, genome editing in the face of intensive agriculture, and the potential risks are only compared with this highly perturbant system. So when we talk about biodiversity or genome editing to enhance biodiversity, when we talk about genome editing in general, we can have a very long list of products. You can name the trait, you can name the application. There are raft of scientists telling we can solve all the problem in the world with genome editing, and this is true for many cases. But when we talk about contributing to biodiversity, well, I just select few cases which has some proof of concept and can serve to do some risk analysis. For example, I choose 3 examples for which the use of genome editing can enhance diversity, and 1 example for which the use of genome editing may result is not well managed in loss of diversity. And I'm talking in all the cases only agricultural diversity and only diversity for crop species. So when I talk about risk analysis, we need to have also a framework, and the common framework to analyzing genome editing application and for decision making in genome editing application is to balance the potential benefit and the potential harms of this development through genome editing. And these benefits are regard in relation to people or to the environment, also a regard in terms of the need, the relevance, the alternative approaches, and the comparative benefit when we compare them. But as I said before, and you can check them in the policy document that are circulating in the European Union. Yesterday, it was out the consultation from the UK. The only reference system used today is the intensive agricultural system as dominant system. And if we compare with this, I will show you later, all the potential damage, risk, harms associated with genome editing are underestimated because the situation under intensive agriculture is really, really bad. So, and needless to say, we talk about people, but we're just talking about some dimensions of public health, or we talk about the health of the environment, and we neglect information as Ruben presented today about ecosystem functioning. So we are using a frame of analysis of risk benefit that is ignoring many dimensions, maybe not in an intentional way, but because simply we working in our scientific over-specialized bubbles, we don't have access to this information that Ruben presented today. We didn't have access to the information presented by Dr. Ceccarelli, and we are not able to use them as alternative reference systems. One of the uses that has been documented for which there is proof of concept is the redomestication of wild varieties. This is an example in tomato, but there is also some proof of concept in potatoes and other species. And what is behind this is using the knowledge we have about domestication genes, manipulate them through genome editing, gene editing, but at the same time, take advantage of the genetic diversity of the wild crop relative. So the idea is that we can take a wild tomato, for example, and take advantage of all its genetic diversity, which may be, for example, made it more resilient to climate change, or more resistant to disease, or with better nutritional quality. It's not the case, but could be for other species. And exploit this genetic diversity, integrate them into breeding program, but at the same time, manipulate the domestication genes to accelerate the domestication process using genome editing. Other application, which is very discussed nowadays in the US, is the redomestication of lost crops, ancient native crops that were lost during the domestication process. Retake them, find them in germplasm, and start redomesticating them, because this can offer a wider offer of nutritional crops to minority populations. Another application also related with the genetic diversity of wild relatives is enhancing genetic diversity. One of this example, I like it very much because show a possible transition, combination, complementation between classical, not so classical, because use molecular markers, breeding with wild species complemented with genome editing. This is a project called iBridges, which was developed at IRD and CIAT in Colombia, which used the genetic diversity of one African cultivated rice species called Oryza glaberrima to introgress genes and characteristics into Oryza sativa. And they were able to break the sterility barriers between the two species and start crossing, then incorporating the genetic diversity. They even were very successful in identifying some introgressors like with resistance to some disease in Argentina, but this was a very long, tedious, expensive process. And now with the use of genome editing and the previous knowledge, we can do the same process by going directly to the S1 gene, break the breeding barriers between the two species, and exploit the genetic diversity of Oryza glaberrima to bring into the O. sativa germplasm. Another example is the use of genome editing for generating allelic variation. This is a project that is being carried on in CIAT, in Arcad, and it's not for application goals, it's just for functional studies, but they demonstrated that we can generate targeted multiple mutations in different genes using the different technologies available, and this will enrich the allelic variation, and we can later characterize these variants to try to identify accessions, genetic material with characteristic we appreciate. And in these three examples, the value add by genome editing is accelerating the process, making it faster, making it cheaper, making it maybe precise. And as I said before, when we use a reference system, for example, in this case, when we analyze risks and benefits for the use of genome editing, we analyze the option offered by classical or conventional breeding. For example, if we talk about off-target effect, we would tend to say, well, the probability of off-target effect is less if we use genome editing than when we do interspecific breeding, because there is all the genetic linkage, drag, etc. So, and we are not measuring it. So genome editing comparatively with this system is better. Maybe the more concerning application, we are talking nowadays about genome editing in agricultural context and for agricultural transition, is the use of gene drive for weed control. Gene drive also are called selfish genetic element. They violate the inheritance laws, Mendelian laws, and can be passed in distortionate ways into the progeny. And they have been offered as a system to reproduce, multiplicate allelic or genes within a population, and they can be used, for example, for the control of malaria, dengue, and other infectious disease. In the case of plants, they have been proposed as a strategy to control weeds, by reducing, for example, the sensitivity to, increasing the sensitivity, to herbicide, or by generating male sterility in the population and then bias the gender proportion into the population. For this application, there is no proof of concept, but there is a lot of modeling studies, for example, with Amaranthus palmieri, which is a very aggressive weed in North America, because there are kind of constrained application to determined by biological attributes. But even if not happening now, there is a lot of talking that this may be happen in the future. And this kind of applications have more risk implicated compared with the previous three ones that I mentioned. So as I said, when we are analyzing these examples, we can do this decision-making framework. We can use this framework. But for every one of the factors we are looking at into the equation, we have to recognize that we as scientists, maybe within one specialization or maybe as a whole, we are not able to appreciate all the dimension of the risk implication. So if I'm going to simplify my risk analysis, I can say that the three first examples are less risky than conventional. Breeding, for example, because they are faster, cheaper, is better. Maybe the last one, I will say that there will be a moratorium, but this is very biased. So there are many factors that has to be considered when we talk about genome editing and when we have to do this decision making, and even if we want to apply a case-by-case approach. Biosafety is a factor. We know we have the instrument to measure it, but this instrument, even if they are in use for a long time, they are not inclusive of all the dimensions. For example, the ecological functioning of ecosystem, for example. We need to analyze relevance or need, but not just in face of the dominant production system, but all the alternatives you were talking today here, how use agrobiodiversity. We need to include also some notions of societal issues, justice, who has access to the technology, who has the right to change trade, who will benefit for these changes. We need to take into account moral conflict, and we need to also be aware that there may be intentions or possibilities of dual use. So what is happening today, and I think this is the most exciting of this moment, and all of you as scientists in the agrobiodiversity field can contribute, is that there is a review of all the instruments, the framework. There is an open consultation in France and in other countries to review and update different framework and guidelines, update the policies, create new policies, adapting ecological risk to other frameworks. In this discussion, the Convention on Biological Diversity is implicated, and the UCN. Also, and this AGAP in CIRAD is very pioneering. There is talk about creating new governance model for these genomic technologies, for genome editing, move towards more inclusive governance models, where responsible research and innovation is an important aspect, and the inclusion of ethical, moral, and social considerations. Also, there is a very interesting proposition of harmonizing at the international level the decisions that have been taken in different countries. There is a proposal for creating an international body similar to the IPCC to centralize decisions, accountability, and databases, information about genomic editing, with the creation of environmental genetic editing bodies, control of which genetic edited organisms are being related in different environments. And finally, también a proposal for the time being, when we still don't have a framework for regulating these novel technologies or the use of genome editing technologies is the creation of an international certification model for integrated deliberation in the way that different sectors are informing about what is happening at the international level in different countries and the local levels as well, supporting and democratizing information access and open information system for decision making. Thank you.
Thank you very much. So on this very hot and controversial topic, I guess we will have a number of questions. If not, I can start. I think there is some point which to me is lacking. It's intellectual property rights.
Yes.
And I would say we could discuss about the wealth and the good and the bad about ecologically speaking or for farmers, but the main point is
yes,
who is going to have
exactly
the right. And if you remember this this trial in the Canada concerning canola, which was modified, and farmers, do you remember this Petcus trial?
Mm.
And it was against Monsanto, and he lost, and he lost the right to cultivate his own variety.
Yes.
So what is your point? Why not putting this property right in this frame?
I didn't put it explicitly, but for me it's including the justice and property. I forgot to put it, but it's very important. We have been discussing about this.
We may say first that no patenting. We may say that first.
Yeah, but yes, but this patenting system is present also in the intensive access to varieties. You know, patents are present in every business. So this is a very
not
hot
in Europe
topic.
Concerning seeds.
Yeah,
so
but I'm not in favor of patenting. I know it's an issue, and the framework has to consider it. But the solution for this issue, we have been discussing it a lot, is not a very obvious one. You say no patent, but maybe you will have sectors contesting this no patent policy.
I would say that as a citizen, and for what we have seen before, I would say for protecting biodiversity for the old varieties,
yes,
you need to say no patenting because no extended rights on the diversity. I would say we do have examples. So, okay, but it's just my point of view. I don't want you to make you upset. Any question on these very, very hot topics? We have one from Salvatore also after this one.
So thank you so much. So interesting. I was wondering if in this first use of using gene editing for domesticating de novo wild species, so that's based more or less on the concept that domestication traits are few and simple, and then you have all the background for quantitative traits in the wilds. And that may be true for some traits, but I don't know for all traits. So I was thinking, for instance, on the so complex pattern of domestication that this morning we saw about pearl millet and lots of integration with the wilds while pearl millet was traveling through Africa and all that. And so I don't know, do you think it is that simple, like getting only a few loss of function traits into a wild species, and then how do you avoid bottlenecks that way? Because that's a strong bottleneck as well. So
yeah, no, I don't think that is that simple. I forgot to say I don't work in this area. I'm not doing redomestication. This is one of the examples that apply when we talk about diversity and the management of diversity. Of course, these have many drawbacks, and people also will highlight some advantages for some species, not for others. Because the domestication process we saw during different talks today is very complex. And also there are undesirable traits in wild relatives and are not already identified at the genetical level, for example. There are ecological functions of those undesirable traits, and we don't understand today. And there is also the interaction, as you showed, with the microbiological community. It's an interaction, interspecific interaction that we are not taking really into account in depth when we are talking about impact at the ecosystem level or at the species level, etc.
Salvatore, do you hear me? Can you get your mic open and ask your question, please?
Yes. Well, my first question was similar to the previous one, but more related to the actual breeding. In other words, how can gene editing cope with quantitative traits, which are the majority of traits?
Yes.
And today we also know that several resistance traits are controlled by a number of QTL on different chromosomes. My second question, if I can ask, from an evolutionary point of view, an evolutionist will probably object that the solution offered by gene editing is not basically different from a biological point of view from the solution offered by the OGM. In other words, is a solution which is so simple, which is within the evolutionary capacity of the organism that we intend to control. So we start again the usual loop, a resistance plant, then the insect of the disease of the weed will evolve, and then we need another one, and then another one. And then, of course, the issue of intellectual property will come.
Sorry, Salvatore, can you make it short, please?
I finished.
I think you are right. Genome editing is actually suitable for single gene traits. It's easier to use genome editing for this kind of trait. There has been some advancement trying to influence quantitative traits and generating, for example, allelic variation for continuous expression of a characteristic. But it's true, it's more difficult manipulate complex quantitative traits, which are the majority of the traits when we are talking about yield, stress resistance, environmental adaptation, etc., are quantitative traits. So there is challenge to improve the technologies to use in this context. But again, I'm not advocating for the use of genome editing. I say this is an option, maybe it's a complementary option for many other approach. Maybe it's not the right one. Maybe we need to look at what we were talking today about agroecology, evolutionary breeding, functional of ecosystem, to try to find a better and more sustainable solution for transition of our agro-system. But if we are considering genome editing, we have to consider these different dimensions, and we have to be active, participating in the debate that are currently taking on the way.
Okay, it's a little, because the time is elapsing short, and it will be the last question.
Yes,
So
thank
Celine,
you,
please.
Thaura. Just to carry on the same issue, basically, I mean, there is a huge discrepancy between how things are presented as simple, cheap, and fast, and the reality of the knowledge that we are completely lacking to make it beyond the simpleness of the use of the tool. Actually, we don't know almost anything on how the knowledge that underlie that would make it even useful. This discrepancy, I mean, is very interesting to also observe, because the narrative is that it will benefit immediately for all kind of problems that has been already created by one kind of agriculture, and that we will use that kind of tool to fix it. So I think it's interesting to, I mean, to have also your view on this kind of discrepancy between the narrative and the technical difficulty that is behind.
Yeah, unfortunately, as I said at the beginning in my introduction, one of the handicap of this analysis within the biotechnology community is comparing the scenarios only with the actual system. This is a reality. You go to the old policy making document, people is comparing what will happen if you use genome editing or if you don't use it when we are implementing intensive agriculture. If this is the system, then you can maybe increase the value of genome editing. But in this context, we are discussing today during the whole day with other options, agroecology, agrobiodiversity, all the things that were mentioned and then just mentioned before. The reference is different, and maybe the value of this, not maybe, surely the value of this genome editing will position it different as well. So the message is, well, genome editing offers some options, but we have to analyze its value when we compare with these many plurality other options that are available.
Thank you very much, Tara. I think that your message has been transmitted and heard and sounded clear. So the debate has already started today. So I would like to thank all the people that gave us a talk this afternoon. I think that we can applaud for them. And I think that Alexandre will make a brief summary of it.
Briefly, yeah, exactly, because we heard a lot of very interesting contribution this afternoon. So we had this view of those presentation to call toward future transition as finally actually as inspiration to pave the way for Arcad community scientific strategy. So we heard Ruben who spoke of crops' contribution to ecosystem processes and linking crop traits and soil functioning. And we, as a case study, we had Salvatore Ceccarelli talking about agroecology and evolutionary plant breeding programs implanted in Italy and elsewhere, and the benefit of it for crops evolving with their natural environment, the network of farmers linking, working on it, and offering pest resistance and weeds control, for example. And then we had Carl Dimmerer from Pennsylvania University talking us about how to take into account cultural and food aspects to understand the importance of crop diversity. Talk a lot about nutrition consideration. And finally, Tara, on this really very controversial subject on GE, genetic editing, talking about evaluating new genetic technology, potential benefit and harms for environment and people, and opening on governance issues. So thank you very much to all of you and for all the questions, interesting questions this afternoon. We are half an hour late now, but I just want to give the floor to Joël Ronfort, which will explain you the tomorrow day program.
So thank you, Alexander. Can you hear me? Yes. I just pick a few minutes. On behalf of the organizing committee to close this session and the first day of these meetings. So it was a pleasure to be here today, and we would like to thank all the speakers, also the session leaders and moderators who have contributed today to the presentation and scientific exchanges. We hope you enjoy this session, whether you participated online or in person. Today we got a comprehensive overview of the different dimensions and scale we address when studying crop diversity, so morphological, genetic, social, ethnological, and ecological dimensions. And from this picture, it appears that crop diversity is a fundamental resource for agroecological transition, a key element to adapt agriculture to global changes, and a core component of interactions between science and societies. So to promote these positive effects of diversity, we'll still need to improve our understanding of the main drivers of crop diversity dynamics and adaptation, and we will continue to work on these research teams. This also opens new scientific challenges, and we want to address some of them in the future Arcad project. For instance, we aim to develop study at the community level to understand how plants interact with their neighbors, and how we can introduce diversity in the fields. We also wish to develop more systemic view of crop diversity, taking into account biological factors, social and political factors together. We want to move towards predictive biology to predict crop and crop-wide relatives with vulnerability, and to develop more dynamic and inclusive management strategies. So for this seminar, we decided to invite most researchers from outside Arcad. We hope this will plant seeds for future collaboration, and will help us to open the Arcad project to a larger scientific community. We want to develop research increasing the combination of different scientific disciplines and crop diversity dimensions, and allowing studies at the interface between natural and cultivated environments, between research and crop diversity management. So we also want to open Arcad for its technological aspects and to the other actors involved in the management and use of crop diversity. This is why tomorrow we will be dedicated to the presentation of the technological facilities available in the new Arcad building, and to the development of synergies between research, crop diversity management and training through interacting sessions involving local, national and international academic and non-academic partners. So this second day, tomorrow, will be held in French in the Amphitheatre Louis Malassi in the Agropolis International Building. So we hope we can see you again tomorrow, and we thank you again and wish you a good evening. So, thank you.